Okay, so we'll mute. Go ahead.
Ready? Okay. So, hi everyone. Our group are going to present functional connectivity of fMRI.
So functional MRI is the type of brain scanning technology that let us see which part of the brain are active and when the person is doing tasks such as resting. And this activity shows through the changes in blood oxygen level dependent fluctuations.
Let's see if this works. Okay.
Hello? Okay.
Good.
Yeah. So blood oxygen level dependent fluctuations, which is the blood fluctuations.
Oh yeah, so just one thing that we don't know why the color is, there's a problem with the color scheme, so
Yeah.
just everyone, I mean, we figured this out following the last lectures.
Was that right?
Yeah, there's
How long has
something
it been
wrong
like
with the
that?
color. I don't know when it changed, but just do your best.
Yeah, just
like a filter that drops in front of the projector or
It's
whatever.
strange, right?
We'll have to tell Doug.
Like blooming.
Yeah.
Yeah,
Yeah.
all blue, we're all blue.
Okay.
Okay. So the both signals fluctuates as brain activity increases or decreases. And now when the researchers want to understand how the brain functions on a broader scale, like which areas work together or just function similarly. And they look for regions in the brain that show similar both fluctuations at the same time, indicating that these regions are possibly working together or responding to the same task. And the functional connectivity helps in identifying and mapping the various brain networks, such as the default mode network, sensory motor network, and attention networks. And these networks are crucial for understanding how the normal brain processes, such as the memory, attention, and the consciousness function. And we use Nylon and Python library to perform the most of our fMRI analysis. And we use the data from OpenNeural. This is a task-specific fMRI dataset and involves in children and young adults watching movies. Is it? Oh. So adults and children watching movies. And we use the data to compare 4 popular methods to extract the signals and construct the connectome, which is the covariance versus the sparse inverse covariance estimation, and the independent components analysis versus the dictionary learning. So the time series is one of the analysis method that captures the intensity of the both signals at each time point during the fMRI scan, and it shows how the brain's activity changes in a specific region over time. And comparing the time series of the two regions of the brain regions reveals how their activities are related. And the brain can be divided into distinct regions, and each can be extracted for 6 representative signals. So here we use the Harvard-Oxford Atlas for different brain regions. And then we use the processing method in the Nylon to extract the signals on the isolation. And this method allows us to input the fMRI data and obtain this time series for each defined region. So once we have the time series data, we can get a correlation matrix that represent the functional connectome. And this matrix shows the relationship between the different brain regions. As you can see, the x-axis and the y-axis shows the same functional brain regions. And each cell's color intensity reflects the strength of the connection between the two regions. So positive values, which should be red, but is blue here, and indicates the most correlated activity. And the negative values, which should be blue, indicates the inverse relationship. So this matrix visualizes the functional connections between all brain regions. And in the left figure, the confound regressors are added during the process, which is important to reduce the noise and improve the accuracy of our figure. And without adding the confound regressors, in other words, if the confound is not removed, which is shown in the right figure, the matrix will contain significant noise, making it harder for us to read.
I will discussing an essential method used in extracting functional connectivities from the fMRI data, which is the sparse inverse covariance estimation, and compare it to the covariance estimation. So Spear's inverse covariance estimation is used to extract functional connectivity by only focusing on the direct interactions between the brain regions. It eliminates the indirect effects and emphasizes sparsity, making the result network easier for researchers to interpret. Most of the brain regions do not directly interact with each other, so we introduce sparsity constraint to capture the biological realities, like what's the directions and pathways we want to measure. The method optimizes a precise matrix that isolates conditional dependency between the regions. From the equation of the Spear's inverse covariance, we can also know there's a L1 regularization, which enforces sparsity and removes the weaker and less meaningful connections. On the other hand, we can see the covariance matrix. It's a widely used basic matrix that calculates the relationship between the brain regions as well. But we can see it here, it only calculates a covariance between two variables, x and y, and it includes the both direct and indirect relationships, so it's more difficult to interpret. From the graph here, we can see the two matrices by the two methods here. So by comparing those two matrices, both of them measure the relationship between the time series signal from different brain regions, but we can see like two pictures reveals differently. Actually, we can see the right side one is more blue. Actually, it's more red, and the left side one is like it's more white. This is because the Spear's inverse covariance captures less signals there, and the white means there's no relationship over there. So the matrices is the Spear's matrices, and we can see the signals to very precisely. And by compared to the covariance one, it is more dense, meaning it contains more non-zero entrants. The correlation here is less significant shown. To further illustrate the differences, we can see this slide shows a visualization of how this brain functional connectivity using the two methods there, and each of them represents the connections of the two brains. The covariance networks are highly interconnected, and we can see there on the top graph, it's very dense here, and it's hard for us to identify. But on the other hand, the Spear's inverse covariance network, it's simpler to identify and show fewer connections. We can see that the regions for this connectivity included here, like from this 3 points of view, and we can also see a paper over here because our data is from the subjects are watching the movie. So we expect to see some signals and connectivities between the motor cortex and the posterior parietal cortex and the visual cortex, which is what the brain regions we use during movie watching. And the connection here fits our expectations. As a result, the sparsity is quite important for the functional MR data because it's helped tell the brain network function connectivity like more clearly and precisely.
So now, to identify and characterizing underlying brain network that may not be apparent just by like looking at the raw fMRI data or simple correlation, especially when we don't have like knowledge and information regarding specific target area ahead of time. So we will use this decomposition method like independent component analysis, ICA, which is based on the assumption that the fMRI data can be separated into additive independent component. So when we first get the 4D fMRI data, it considers 3D volume collected over time, forming the 4D data set. Then we will have our matrix that is underlying the mechanism of this ICA method, which is x equal to AX. So the X is the fMRI data we got, and then our goal is to identify S that are maximally independent from each other across the data set. So the ICA output will give us a spatial map and their associated time courses. So this component represents different patterns of brain activity that are spatially distinct but may overlap functionally. So in the left is corresponding to the x equal to ax, and each voxel in this volume represents a point in brane space, and the intensity value of each voxel change over time based on the both signal. So then we create our whole brane mask to extract the time course using the CAN-IC model in this Nelearn Python library. And then we get our data. This is the all IC component. So and then this is the separate IC component. So each color encapsulates a spatial map of brane activity that is independent from the map represented by other colors. So each color region indicates area in the brane where the voxel activity are highly correlated with each other within that component. And we verified that this temporal activation pattern, this time course of the component are not significantly correlated using the mutant information statistical method to show that is indeed independent region to each other. And yeah, and at the end, we also did one dictionary learning to compare with our ICA since they are really close related. So the dictionary learning is just a sparsely based decomposition method, extract the math that are naturally sparse and usually cleaner than the ICA. So because of the space limit, I didn't show all the separate component, but this is the dictionary component result that we got, and is similar to our ICA data, but is more concise and then is more accurate than the ICA. And this is our citation. Thank you.
Talk's open for questions. Any questions? Yes.
Could you clarify for me, so it seems like you took like a couple different approaches in terms of like analyzing things. I just want to clarify. So so you used the atlas-based approach to find functional connectivity with the covariance matrix, and then also used ICA to find subnetworks separately, or did you combine those approaches somehow?
No, it's separately, just two different methods, and they are using the same data set. Okay. Yes. Sorry, didn't clarify that in the ICA because of time limit. Yeah.
There's another question.
How did you all account for, sorry if I missed this, but how did you all account for any confounding variables such as motion when you're watching something or physiological noise? Did y'all normalize or?
Oh, yes. So because the data set is already pre-processing where we get it from, so it basically is already like clean data set is ready to be used. Yeah.
So based on your project, what is the best method to use then?
So let me go back. So I will say it's the ICA because I'm the one doing that part. And then after seeing their results, so that's my opinion because I think it's more clear and it's a popular method, and so that's why it's easier to access because there are a lot of like tutorial on this ICA method. So I think and based on the results, I think is like I didn't do the like what we did here with our covariance connection, but I didn't do it here eventually, but we got this. But I think because we have this independent component at the end, it gives us like it can serve multiple purpose when we are doing like the further study because we have this independent like component, like it can tell us like which region they work together on one thing. Yeah.
Thank you. So let's move on to the next group. There's a question for you on the chat, so you can respond to that over Zoom.
Also we just
Yeah,
type
just
it.
chat.
Oh, okay. Yeah, sounds good.
Where's the next group?
Yes. Okay.
Go for it.
Okay. What is this? Do I say it loud?
Yes. Sorry.
Okay. 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 Oh, shoot.
No, it's fine.
Okay. Okay. So our project involved designing a real-time automated assessment of image quality during MRI scans for optimal image reconstruction.
Why did that echo?
It says it's paused in the screen sharing.
What the heck?
Do I resume?
I guess resume, yeah. Mhm. Maybe I'll just have to reshare my
Or
screen.
slide control, maybe?
No,
Oh,
I think it is the resume.
it's not resuming. Oh.
Are you okay doing full screen?
Yeah, that's
Yeah.
fine.
Okay, we're gonna
And then I
do,
could just do this.
Sorry, guys.
we're gonna try again. Okay.
You can press like escape there.
See it there?
And then
And can we do side by side? Is that
yes.
chill?
And this works. Okay,
Yes.
yeah.
Okay.
Do side by side.
Sorry, guys.
Okay. So our project involved designing a real-time automated assessment of image quality during MRI scans for optimal image reconstruction. So as we know, motion is a problem in MRI. Like we've discussed in class, when a subject moves during an MRI scan, the resulting images will be corrupted with artifacts or blurring effects. So one way we can reduce motion artifacts is by using radial sampling. So radial sampling is a method of acquiring case space where each line that's acquired, which is known as a spoke, passes through the center of case space and forms this star formation here. So typically these spokes are rotated by a mathematically derived golden angle, such that the spokes are evenly distributed and they never repeat one another. So radial sampling is more robust to motion compared to Cartesian sampling. And this is because in Cartesian, we know that the phase encode direction is fixed in one direction, but in radial sampling, the phase encode is in varying directions. So when you have a motion artifact that occurs in the phase encode direction, as they often do, those artifacts will be distributed in varying directions, becoming blurred out and less noticeable. Additionally, because each spoke goes through the center of K space, if there's a motion artifact in one of those spokes, it can be more easily averaged out. So this is an example of the effect of motion during a scan acquired in Cartesian sampling compared to radial sampling. And you can see that in Cartesian, motion causes these ghosting artifacts, whereas in radial sampling, we see the blurring effect that I was just describing. So we can simulate this blurringness, therefore simulating the effect of motion by simply acquiring fewer spokes in K space. And this is because inherently in radial sampling, the center of K space will be oversampled, whereas the areas towards the edges of K space will be more sparsely sampled, which is shown in the blue or that top figure. So if we acquire fewer spokes, we'll have very little information from the edges of K space. And we know that the center of K space gives us general structure information of our image, whereas details such as sharp edges come from the edges of K space. So if we downsample the number of radial spokes, the resulting image will lack those sharp details like edges, but contain structural information and therefore will result in a blurry image. So this is one way we can simulate the effect of motion using radial sampling, which is something we'll discuss more in our methods. So radial sampling is a great way to reduce motion artifacts, but if you're imaging something like the heart and you have to deal with physiological motion, you're gonna have problems. So in cardiac imaging, one technique that's used is acquiring data when a patient is holding their breath. Another technique is gating. So here, an ECG is used to detect the electrical activity of the heart, and then MRI data acquisition is synced with specific phases of the cardiac cycle, which ensures that the images are captured when the heart is at a specific position, such as systole when the heart is contracting, or diastole when it's relaxed. And then you can sum up data for multiple heartbeats over time to obtain better resolution in your final image. So if you consider a cardiac scan where data is collected during a breath hold and ECG gating is used to acquire data at the same point in the cardiac cycle, you can imagine that if the subject has an arrhythmia, is unable to hold their breath, or just moves at some point during the scan, that event could corrupt your data set and result in a suboptimal MR image. And the problem is it's difficult to detect and mitigate those kind of motion issues until after the scan is complete. So our goal was to create a quantitative method for real-time evaluation of MRI scans and to identify those motion events when they occur.
Ideally, this could be used in the future by designing a real-time decision tree that would discern how to proceed with a scan after identifying this event. But for our project, our goal was just to identify this event specifically. So we did so by simulating the motion events that I just described, and then we trained a machine learning model to identify them. So if you remember this slide from class, when we talked about deep learning, this is an example of using a machine learning technique that falls into this first category, and it can be applied for optimal image reconstruction.
So let's talk about the methods. Our data set is from the publicly accessible ACDC dataset, which consists of 150 patients, 10 short axis cardiac slices, and 30 time points across the diastole-systole cycle. In order to produce our corrupt simulated data, we performed 3 adaptations to the raw data, which included reducing the number of radial spokes, like Cassie alluded to, producing an image from half the spokes in systole and half the spokes from diastole, and then adding an affine transformation in the image space. So for each data point, we produce a Fourier case space from 1 patient, 1 plane, and 1 time point in diastole. Then using variable number of golden angle radial case space spokes, we produce a series of images with the inverse Fourier transform. Using the non-uniform FFT, we use 10, 20, 30, 40, 50, and 200 radial spokes with the increased number of spokes producing the better quality images. By having images produced from a lower number of spokes, we can simulate corrupted data. Then for our second adaptation to the original data, we apply an affine transformation to the images produced from the variable number of radial spokes. The first transformation is a 30-degree rotation, and the second is a 200% zoom. Although these are extreme transformations, they can simulate corrupt images produced due to motion within the scan. We also produce data with no affine transformation so that we can have images just with a reduced number of radial spokes. Lastly, to adapt our original data and simulate motion corrupt images, we acquire the Fourier space from 1 patient, 1 plane, and 1 time point in diastole. Then we also acquire the Fourier space from the same patient and plane, but a different time point in the cardiac cycle, this time being from systole to simulate incorrect cardiac gating. Again, we use variable number of radial spokes, but this time we take 50% of the spokes from the Fourier space in diastole and 50% of the spokes from the Fourier space in systole. This will produce a superimposed image of the heart from 2 different points in the heart cycle, simulating the instance of incorrect cardiac gating during the scan progression. Next, we need to score the degree of corruption for the data we produced. Again, the corrupt simulated images could include a reduced number of radial spokes, an added affine transformation, a superimposition of 2 time points in the cardiac cycle, or a combination of the 3 adaptations. We compared each image to the corresponding image produced by the same patient and plane with 1 time point in diastole, 200 radial spokes, and no affine transformation added. To compare each image, we utilize the Structural Similarity Index Measure, or SSIM, which outputs a 0 value for 2 completely dissimilar images and a 1 value for identical images. Finally, we chose the ResNet 18 learning model to recognize the corruption in images, since this model inputs images with associated float values and outputs estimated labels between 0 and 1, 0 being most dissimilar and 1 being identical. For our training set, we used over 600,000 images. Then for our testing set, we used over 160,000 images.
And so here we have our results. And so this graph is comparing the SSIM scores to the labels that our ResNet 18 learning model was able to estimate. And so as we said, the SSIM scores are the ground truths that we trained the model for, and they essentially quantify the image quality to a score between 0 and 1. And so if our model works correctly, we should then see a directly proportional linear relationship between the SSIM scores and the estimated labels. And so as you can see here in our graph, the equation for our regression line is shown up top in blue, or I think it's red, and with the flip-flop. And so it's very close to the y equals x line that would mean that our model perfectly estimates the SSIM scores. And so we achieve this with an R value of about 0.93. And so our model does actually slightly underestimate the SSIM scores, but we can use that knowledge to our benefit if this were ever to have an actual clinical application. And so to further assess the agreement between SSIM and estimated scores, we assigned each image quality to bins with ranges of 0.25 from 0 to 1. And so in those four bins, we made this confusion matrix, and it has accuracy per quality or per accuracy per category. We got like about 98% across all the bins. So I think that is a really good score. And here we have some example images of our test data. And so those scores on the top are the SSIM scores that are like the ground truth SSIM scores that we were trying to predict. And the numbers on the bottom in this figure are the scores that our model was able to predict with the ResNet 18 learning stuff. And so as we can see here, once there is a motion corrupting event during the scan, our model is able to detect it, and from there, like in the future, like a decision could be made about what to do after the motion corrupting event. And so some other methods to like our machine learning model is that or some other methods to address the corruption of motion, the motion corrupting artifacts. So we have like parallel imaging, which we learned about in class. So that those are techniques such as Sense and Grappa that use multiple coils with their own spatial sensitivity profiles to reconstruct an image. And so the way that methods such as parallel imaging and multiband imaging actually address motion artifacts is just by reducing the time of the scan. So there's less likelihood that the patient will move or that there will be a corruption. And additionally, we can use different motion tracking techniques, such as navigator echoes, to detect different changes in the coil that would indicate motion. And like all of these methods, as the last bullet point here says, take advantage of the coil sensitivities in some way. And finally, we can do retrospective motion correction, where artifacts in the image attributed to motion can be corrected after the image is taken. And so oftentimes, though, those will require very intense algorithms capable of reconstructing the image in a way to compensate for the motion artifacts. And so in conclusion, we were able to make a ResNet 18 learning model to successfully estimate MRI and SSIM scores, and therefore enabling us to detect motion artifacts in real time during imaging. And in the future, we would like to be able to do like a decision tree to determine whether we should be restarting the scan or continuing and correcting and just like cutting out a time point. Thank you.
Yeah, one from President Beckett.
So.
The next group come
The
up.
real problem often is you've got 8 good heartbeats and 1 bad one. Right? And I couldn't figure out from the way you changed your ground truth SSIM if that case is in there. It seems like you went from
Like this.
spokes, many spokes, with 50% of them being wrong or something. Is that right?
In terms of simulating the data, correct? Here.
You have 50% of your spokes are from an errant beat, 50% from good beats.
Correct.
That's right. So I think this is a great setup in terms of the way you've done it. But I would run it with, like, just adds 1 bad beat.
Mhm.
So versus 11 good beats or something like that.
Mm.
1 bad beat versus 9 beats or something.
Okay.
That's the thing you really need to detect and go back and hit that beat again.
Mm.
Right, so, great.
All right, great job. There's some questions for you on the chat that you could answer those offline,
Okay.
and we'll have
Okay.
next group come up.
Okay.
Okay, thank you.
Thank you. That's fine.
Thanks.
Is this ours?
Yes. No, that's not.
Okay,
Great, okay, go ahead and get started.
sure. So, hello everyone. My name is Uma, and these are my classmates Rohan and Nathan. And today we'll be discussing chemical shift imaging. Okay, so to provide some background, chemical shift imaging is an MRI technique that is able to find the difference in resonance frequencies of fat and water protons to then differentiate tissues based on their fat content. So fat protons spin at a lower frequency compared to water protons due to electron shielding, which led to the term chemical shift effect. And CSI takes images at a specific echo times that then use like phase differences between the fat and water protons to its advantage. So the in phase images are when fat and water protons are in phase, which result in additive signals and its brighter appearance of fatty tissues. The opposed phase images are when fat and water are out of phase, thus canceling and reducing the signals from voxels, which have both the fat and water. So the image that I've put on this slide shows the in phase and the opposed phase images, the brighter being the in phase, and the opposed is the darker one, and it's characterized by the India ink artifact, in which those like dark regions are where the boundaries of tissues mainly composed of fat or water lie. And the images are like the opposed phase images are really useful because you can see the contours to recognize boundaries for organs or lesions. In this case, you can see like a dark outline for where like the bones are. So some applications of chemical imaging include musculoskeletal pathologies, marrow pathologies, fractures, and microscopic fat in tissues. The image shows an example of how CSI can be used to rule out metastatic lesions. So this patient, whose like images are on the screen, came in with back pain, and figure A shows marrow edema, and then B shows a horizontal fracture line, but C and D are the in phase and opposed phase images. So the metastatic lesions are typically what they typically replace normal bone marrow, and normal bone marrow is really rich in fat. So in comparison, the lesions would have significantly less fat signals compared to the normal bone marrow, but this image shows strong fat signal in the opposed phase image. So that ruled out the metastatic lesions and then confirmed the diagnosis of marrow edema associated with benign compression fracture.
So our project will focus on detecting hepatic steatosis in the human body. Hepatic steatosis is essentially the buildup of triglycerides in liver cells, which is commonly associated with chronic liver disease. It can be broken down into two different forms: non-alcoholic fatty liver disease and alcoholic liver disease. And over time, this condition can often contribute to liver failure, cardiovascular disease, and metabolic syndrome. And for testing and diagnosis of this disease, liver biopsy remains the current standard, but due to its invasive methods, it can result in many complications, including physical pain, as well as sampling errors for detection. However, there's been a growing interest in non-invasive techniques such as chemical shift MRI, and so our project simulation will involve utilizing this technique through MATLAB.
So we have two primary components to our MATLAB simulation. One is a bit more visual and one is more mathematical and analytical, which Rohan will talk about after I do. And so for the visual component, it was a little bit weird getting started because my goal was to take a traditional MRI image and augment it so that I can show some of the differences and show the fat content. But through MATLAB alone, this isn't possible because it can only read the specific brightness of individual pixels. So since the liver in the right image is roughly the same shade of gray, there's nothing that MATLAB would be able to do to show those differences. So it meant I had to start with an already acquired image from chemical shift MRI. And then I had to do something to augment it from there. And so I decided to do something similar to what we did in homework 6, question 2, where we took different images of the brain and sparsed out different components, highlighting each one, and then augmenting them from there. And And so we can see that I made 3 sections. So most of the liver stores so much water that most of the signal that shows up is a water signal. So I made everything below 0.57 out on a scale of 1 brightness to be considered water content. And it does include other parts of the body that have similar water signals. But our main focus is on the liver and separating out the portions of the liver. So everything above 0.85 is going to be considered brown fat, which is healthy. And so you can see some fat stores around the body, those small globular regions, as well as the spine is also included as well. But again, most of that information, the brown fat and the spine, we're mostly going to ignore for the purposes of our project. So everything in between those 2 points is going to be considered the white fat, which is the unhealthy fat building up in the liver, causing the disease. And so we can see in the bottom image, there is plenty of fat content showing up that originally was present in the liver and is now showing up separate from the water content. And so from there, having those individual pieces split into different images, we were able to augment each one individually. And so I was able to recreate the original image by just adding back each layer with the original weight. But then I was able to change it a little bit by adding back the white fat layer on top of it, making it a bit more defined. So we can see in the image on the right, the water content of the liver, it appears much darker and has significantly improved contrast between the white fat and the water content of the liver. And additionally, in the original image, the brown fat and the spine appear to be the brightest in the image and draws most of the focus. But then now in my version of the image, it is significantly darker. And now the white fat is the main focus of the image.
Yeah, so to validate our MATLAB images of isolating the water and fat content in the human liver, we use chemical shift frequencies that accurately correspond to both white and brown fat. And so using the equation shown here to determine, we use this equation to determine the frequency shift. And these frequencies were based on previous studies and are commonly used. And so the parameters seen here are the ones we use for the simulation. The field strength represents the magnetic field strength of the MRI scanner. The gamma represents the gyromagnetic ratio for protons, and the delta being the chemical shift in ppm. And so the chemical shift differs per component. So with water having a value of 0, white fat having a ppm of 3.4, and brown fat being slightly less than white fat. And so to ensure that our simulation was properly performed, using these values, we ran a frequency spectrum that displays what frequencies these components are being utilized in. And from the peaks, they accurately represent our expected values of 0 hertz, 347 hertz, and 434 hertz. And based on our image that we used, we also wanted to provide a case-based visualization to compare the low and high frequencies of our image simulation. So in order to acquire this image, a number of functions needed to be performed to generate the case space. To begin, we needed to perform a summation of our time domain signals at 3 time points. Then a 2D Fourier transform was applied to convert the spatial signal into a frequency domain. And then the FF shift function shifts the frequency component with 0 to the center. And then next we use inverse Fourier transform to transform the case-based data back into the spatial domain and reconstruct the image. And then we use image SC to display the case space shown on the right. And from the image, we noticed that a bright center point to represent the low spatial frequencies. However, we did notice a relatively bright peripheral of high spatial frequencies that surround the center, which could signify more detailed edges and finer features within the liver image.
Okay, so some ways that CSI can kind of like go in the future would be first, like expanding applications in musculoskeletal imaging. So being able to differentiate between benign and malignant lesions, or even like characterizing cartilage and soft tissue abnormalities could be useful. Another path could be a quantitative analysis in musculoskeletal imaging. So finding like a standard protocol for like a region of interest, like and then going from there, or even like normalizing some of those signal intensity ratios. So for different like tissues or pathologies. And another path we could go down is like integration of CSI with other imaging technologies, such as diffusion weighted imaging or magnetic resonance spectroscopy. So this would just give us even more information about different tissues, pathologies, and then cellular and microstructures. And then finally, one more way we like CSI could go towards is exploring like novel applications such as neuroimaging. From there, like looking at how different fat deposits can contribute to neurological conditions like Alzheimer's. And then one more would be cardiac imaging and how myocardial fat content can be associated with the risk factor for cardiovascular disease. So here are our references, and then thank you. Does anyone have any questions?
Go ahead. I know that most of the fat in the liver is intracellular, and so generally you can't see it just by thresholding. So I'm wondering where you were showing the spectrum of the frequency shift are you trying to represent it, or I guess a better question would be, is this confined to the liver, or is this across the entire image, including the brown and the white fat that was outside of the liver?
My understanding is that it was around the entire image, but if we had a bit more time or more resources or future directions, it would be to focus primarily on the liver. Because as you can see, like with my images, like right here, this is the spleen, and it's being considered brown fat, which isn't necessarily accurate.
Right, and then there's like the aorta.
Yeah, exactly. Yeah, like there's plenty of stuff that's present that isn't necessarily accurate. So like if we could recreate more layers and have more thresholds to separate those out better, that would definitely be something we can do and have more accurate graphs too.
With this technique, could you potentially just segment the entire liver and then only use the area that's inside that, like maybe with vessel removal?
That would definitely be possible. Yeah, like if we were to just only use portions of the image, that would definitely be something we can do. But for the purposes of our project, we figured this would be sufficient.
Is there another question? Okay, you can ask your question. Can the other group come up? Next group come up while we're waiting. Yeah, go.
Do you know the difference between an alcoholic and a non-alcoholic that has a liver disease with this injury?
Not primarily. They're just classified into two different forms of diseases. I think they do have different quantitative assessments between the two. But with our simulation, with further improvements, we probably could.
Yeah, I think it's mostly used as like a diagnosing the issue responsible so that it can be future addressed rather than like determining the difference in the effects.
So in your simulation, I didn't really quite get what you did. So you said you had 3 different time points. What do you mean by 3 different time points?
So these aren't time points, these are brightness thresholds.
No, go to a later slide, like one of your last slides. Keep going. Yeah, summation across time dimension, 3 time points. Like what? Could you clarify what that means?
That was incorporated into the initial liver image itself. It was part of the simulation using time domain signals. But that was just incorporated to generate the case-based image itself. Because the code used 3 different time points, if I remember correctly. I don't remember which values, but they were used to provide those images of isolated liver or water and fat.
Because as Uma described in the background too, this form of imaging does also incorporate different echo times when imaging too. So I think this is also probably just to reflect that.
Yeah.
Okay, great. Thank you. All right, thanks.
Thank you.
Thank you.
We are now. Yeah.
Great. Okay. Go ahead.
Do we need to record?
No, I'm
no.
recording.
Okay. Minimize that too. Okay. Yeah, there you go. Cool. So, hi, my name is Ping. We're group 11. Today we'll talk about how diffusion magnet, DMRI, specifically diffusion weighted imaging, can provide quantitative, biologically meaningful information about hepatocellular carcinoma microscopic structure. So our focus will be on leveraging these diffusion-based biomarkers, particularly the parent diffusion coefficient, to better understand tumor grading. And in other words, we want to see if the degree of cellularity inferred from restricted diffusion can help distinguish infer from restricted diffusion can help distinguish between lower grade and high grade tumors. So on this slide, we show the concept of an MR voxel, which is basically the smaller three-dimensional measurement unit in our images. So each voxel usually has a solid length of few milliliter and contains millions of water molecules. So within the voxel, we consider there are three key compartments. First is intracellular space. It's the extracellular fluid located between the cells, which allows relatively free movement of water. Then we have intercellular space, the space within the cell membrane. Here, water diffusion is more restricted due to the presence of cytoplasmic organelles, cytoskeleton, and cell boundaries. And also we have intravascular space, the blood vessels present within the voxel, in which water flows as part of the blood. So by combining these signal from all these compartments, diffusion-weighted MRI captures the weighted averages of water moving freely in different microenvironments. So the essence of diffusion-weighted imaging lies in the use of magnetic field gradients. In the pulse sequence, these gradients apply at least twice at different moments, once before and once after. For stationary molecules, these gradients produce a symmetrical effect. The first gradient encodes the phase shift, and the second gradient is used to refocus. Since stationary molecules do not change their spatial position, the net effect after the two gradients is zero, which means that there's no net signal loss. However, suppose a water molecule diffuses from its initial position to another position in short time between the two gradient paths. In this case, the second gradient cannot perfectly realign the initial phase encoding. So this phase mismatch causes a slight signal loss, which is manifested as a measured attenuation of MR signal intensity. The degree of signal loss is therefore directly related to amount of diffusion. So by adjusting the strength and timing of the gradients, we can quantify the degree of water diffusion in tissue. To simplify the complex diffusion process, we commonly use a parameter known as the apparent diffusion coefficient, or ADC. The ADC is not a true diffusion coefficient in a physical chemistry sense, because water molecule in biological tissue is not diffusing in a simple manner. ADC values reflect how fast water molecules diffuse within tissue and can be influenced by cell size, density. And in cancer imaging, one key pathological feature that often distinguish aggressive high-grade tumors from their low-grade counterparts is cell density. So high-grade lesions frequently show densely packed malignant cells that limits the extracellular space through which water molecules can diffuse. So the decreased restriction translates into a measurable decrease in ADC. If we can symmetrically correlate ADC value with pathologically confirmed tumor grades, then ADC becomes a clinically actionable biomarker.
So to get some conceptual idea of what's going on here, we need to understand the background in which these tumors are growing. So this is a histological picture of a normal liver, and I want you to take note that there's relatively evenly spaced cells and nuclei. There's a lot of free space in there. But keep this in your mind when you look at these next images. So the next thing we want to look at is a poorly differentiated HCC. And what you see is we have these large basophilic, like almost osteoblast-like tumor cells. The actual boundaries of the hepatocytes are no longer visible anymore. Instead, these tumor cells are just living in these large hepatic plates. And so we would imagine that the water molecules in this sort of environment are going to be bouncing off of these large tumor cells, and they're going to be getting stuck in the crevices. So there's going to be more restricted diffusion in this kind of environment. But that's kind of making things a little bit too easy on ourselves, because obviously a poorly differentiated HCC by definition is going to be very different from the surrounding tissues. On the right-hand side here, you see a well-differentiated HCC. And besides the fact that we have like these sort of trabecular structures moving through, the actual spacing of the tumor cells relative to the nuclei and the spacing of the hepatocytes is relatively similar. So what we would expect is that there might not be as much difference in the diffusion of a well-differentiated HCC versus a normal liver. And so what we've found is that in the literature is that histopathologically confirmed HCC has a higher ADC in a well-differentiated tumor than in a moderately or a poorly differentiated tumor. And what this allows us to do is use the apparent diffusion coefficient to grade a tumor. So one thing we do know is that a poorly differentiated tumor is generally a more aggressive tumor. So the ADC allows us to assess the sort of treatment response that is necessary for each type of tumor. So specifically, one of the questions that we wanted to ask is how do we optimize a sequence to look at this type of data? So one of the things that we have to consider is the changes in the TRs and the TEs, the echo train length for a body image versus a brain. If you look up diffusion imaging, 90% of it is in the brain. And there's a reason for that. It's actually quite a bit easier to do in the brain. And so you have to take into account more complicated structures when we start designing these sequences to look at the body. So what we have here is we have an equation for looking at the relative signal to noise ratio. The difference from the normal equation that we use to look at SNR here is that now we have to take into account the amount of time that we are firing these diffusion gradients. So actually, if you can go back real quick, I have a table here showing how on average, the T2 structures in the liver are shorter than in the brain, and that's important. So what we did is we plotted this equation with various tissue T2s. And one thing I want you to note here is that the red dots are indicating the relative maximum signal to noise ratio of these different T2 values. And so what you can appreciate is that as the T2 value gets shorter and shorter and shorter, to achieve the peak SNR, our echo train length also needs to get shorter. And this kind of makes sense if you think about it, because if our echo train length is too short, then we spend all of our time firing these diffusion gradients and diffusion weighting the image, but we're not actually collecting any signal. And if the echo train length is too long, well, then our T2 has relaxed away. And now all we're picking up with the latter part of the echo train is noise. So the important takeaway here is that we can't just simply use a normal brain setting for body. We definitely need to adjust for this shorter echo train length, and that can be a actual hardware limitation on the system because, of course, as we all know, these magnets have limits to their gradients and their TRs and their TEs, and we might not be able to achieve a good signal if the T2 value of the target is too short. So the other thing that we looked at was the choice of B values. So what you'll normally see when you're acquiring a diffusion weighted image is you'll see a series of B values, normally 2, sometimes 3, and you'll have a baseline value B equals 0, and then you'll have a high B value somewhere in the 500 to 800, maybe even 1,000 range. So in the body, we cannot do that because of the properties of the vascular perfusion. So if you look at this graph on the right, the thin dashed line is the signal attenuation represented as signal versus B value. And you can see that it starts out very high and it very quickly attenuates throughout this low B value range. And then it becomes roughly linear afterwards. Now, if we just take a normal ADC map interpretation of this, you get the thick dashed line that we see there. And the slope of that line is the apparent diffusion coefficient. But what we actually want to be doing is only sampling the range where the values are linear, because if we're not sampling over a linear range, we're not actually getting a good representation of the diffusion. The reason this happens is it's not exact. It's still debated in the literature, but it's basically capillary vascular diffusion. And if you see in the image below, all of the vessels within the liver are showing up bright. And if you remember from looking at the DWI images, bright normally means restricted diffusion, but these are blood vessels. The diffusion is not that restricted at all. And if you're trying to look for a tumor on here, that creates a very complicated and messy image because now you're seeing all these bright spots throughout the liver. So we have to take a B value that is higher than 0. It's ideally higher than 50, where we have already gotten past this area of very, very fast attenuation. So when we're optimizing a sequence for ADC for looking at hepatocellular carcinoma and cell microstructure, we need to adjust these B values just like we need to adjust the echo train length.
So in future direction, here I want to emphasize on combining DWI with other MRI contrast agents. So, or just like contrast. So pairing ADC maps with a perfusion-weighted imaging, MR spectroscopy could have composite biomarkers that integrate cellularity with vascularity or even metabolic activity and iron content. So this image demonstrates the measurements of imaging biomarkers as apparent diffusion coefficient and stiffness in tumor and peritumor area. And here we have T2-weighted the superparamagnetic ion oxide nanoparticle. So these contrast agent can be engineered to target and highlight specific cell populations such as T lymphocytes, immunoregulatory cells, or even particular cancer cell subtypes. So by simultaneously collecting data on cellular density from ADC, vascular supply and perfusion from MR spectroscopy, and iron-related change from SWI, we can build a novel imaging fingerprint of a tumor. So to conclude that, identifying tumor microstructure with diffusion MRI is a useful tool for risk stratifying patients with HCC. ADC maps allow quantitative non-invasive assessment of tumor cellularity. Optimizing DWI sequence for body imaging require additional consideration to the diversity of structures involved. This is our references. Thank you.
I have time for one question. Could the next group come up in the meantime? Yeah, so what sort of gradient amplitudes do you for your simulation? I mean, obviously, if the gradient can be larger, then
Yeah, so we just chose to use standard 3T. So if you see here, the equation itself doesn't actually depend on the B value. So the equation is you just have the MZ, which is the return magnetization, and then you have the T differential, which was the gradient firing time. So we used a standard 50 millisecond as the gradient timing.
right.
And then the T2 values were the tissue T2s, and then we graphed T data. So one thing is that if you wanted to represent just a single tissue, you would want to normalize this by the T2 values because you can see that the relative SNRs are not the same. And in reality, those probably would be the same. You wouldn't have a big change in SNR just by changing the T2 of what you're imaging, but just to be able to display them and show the effect of the maximum value changing, we did not normalize them there.
Okay, great. Thank you.
Yeah.
Let's have a next one.
Hi everyone, we are group 15. My name is Aining, and my group members are Andrew and Alejandra. Today we're presenting our project about ramp sampling. Sorry. In homework 5, we explored the design of echoplanar imaging, the simulated gradient waveform for GX as a rectangular shape, and the sampling point in K-space are evenly spaced. EPI represents one method of acquiring data characterized by its unique K-space trajectory. If alternative techniques used different gradient designs, we would get varied K-space trajectory and imaging properties. Today, we'll discuss another method called ramp sampling. In a readout gradient waveform, we notice the trapezoidal shape gradient lobes. This trapezoidal shape is commonly seen in real-world MRI system rather than an idealized rectangular shape. In traditional API, data is only collected during the flat plateau portion of the gradient, not during the ramp. However, in ramp sampling, data is collected in both ramp and flat portions. The k-space samples are nonlinear during the gradient ramps. When data is collected only during the plateau of the trapezoid, the k-space samples are linearly related to the time domain signal, but ramp sampling introduces new efficiency and complexity. Here we will compare ramp and non-ramp sampling from a mathematical perspective. T1 and T2 represent the beginning and the ending point of the ramp portions in a gradient waveform. When data is acquired continuously, including the ramp portions, the readout k-space trajectory follows the equation shown here. The slew rate defines how quickly the gradient changes and determines the slope of the ramp. In contrast, non-ramp API only collects data during the flat portion of the gradient, where GX remain constant. These fundamental differences impact the efficiency of k-space, traversal, and echo spacing. An application of ramp sampling is facilitating the real-time metabolic imaging. The study aims to track the hyperpolarized carbon-13 pyruvate and its conversion into lactate. A major challenge is that HP signal fades quickly, given short time to collect imaging data. So they combine the symmetric API with ramp sampling and partial Fourier reconstruction with reference scan acquired in the proton channel. They use spatial RF pulse to excite only the desired metabolites by selectively exciting a single resonance. The symmetric API collects data during both the positive and negative parts of the gradient cycle, and ramp sampling collects data continuously, and the echo time is substantially reduced and the SNR is increased. In this project, we're going to simulate the ramp sampling and compare different reconstruction methods.
So the way that we looked at ramp sampling was we had a MRI brain slice, and we took the Fourier to get the k-space, and then we ramp sampled that k-space. And once we reconstruct the image, we get the image on the left. And you can see that you have like this big gap in the center. And that's probably due to the fact that with ramp sampling, all your points are concentrated on the edges, with the center having more sparsely sampled k-space points, and that leads to a lot of aliasing. So with ramp sampling, you need to have some considerations with reconstructing the image. So one of the things that you need to do for reconstructing it is regridding that k-space to make sure that it's linear, so that we can actually take the inverse Fourier and get the correct image back. So here you can see the ramp sampled k-space where the change in kx is actually changing, so it's not linear. And then with the non-ramp, you have a linear correlation. So once we regrid it, the way that we regridded this one was by interpolating the values for kx, and we were able to get that linear correlation again. So the k-space trajectory that we designed for looking at the image was the following, where I assigned 20% of the points to be part of this linear increase in the beginning. So you can see that there's more points on the edges, because as we're increasing the change in kx, the points start to spread out until we reach that center where the spacing is even. And then on the opposite side, the reverse is true, where we start going from large spacing to smaller spacing. And from this sort of k-space sampling, we can regrid by interpolating uniform space values. So it is kind of like making up maybe data because we are interpolating, but it leads to having a uniform change in kx. So this was just another way that I visualized it, where now we're just looking at a row and then the change in kx as you go through the row. So you can see for the ramp sampled, you start small and then it increases till it reaches that plateau where it's constant. And then on the other side, it goes from large to small, whereas the non-ramp sampled and the regridded are basically aligned because the change in Kx is uniform across Kx. So once we do this regridding, you can see that we get a reconstructed image that is a little bit blurrier, but we don't have all that aliasing that we had in the previous image that I was showing. You can see the reconstructed image with the uniform sampling obviously has better resolution, but if the signal is going quick, then being able to reconstruct from ramp would be beneficial.
Okay, so in order to simulate ramp sampling, like a standard EPI trajectory without regrading and interpolating, since like mentioned before, we're sampling an image that's already been sampled instead of an actual object. We designed our K-space trajectory like this. We needed to condense the sampling towards the edges of the K-space where the GX gradient is experiencing that ramp effect, and then gradually getting less dense towards the center of K-space. Some design considerations we had to think about were keeping it symmetric, that way the KX gradient was even. And we also had to make sure the dimensions met the image of 208 by 300. So as stated earlier, the method of sampling for EPI is pretty challenging because it results in a lot of aliasing, even with like linear or cubic interpolation. The two images on the left were constructed with that previous slide's trajectory. And the one on the far left also went through 1,000 iterations of compressed sensing to try to fill in that missing K-space that we're seeing. As we can definitely see, we have higher details like on the edges of the image, like the left and the right. We can start to sort of see like a contrast between gray and white matter take shape. But the bottom and the top are very blurry and undersampled because of that missing K-space in the middle. And the middle of K-space is where the most valuable information is. So the images on the right were generated with the same trajectory, but the middle of the sampling was more dense and a lot more samples. So the center of K-space was more highly sampled, leading to much better reconstruction. And after compressed sensing, it's like an almost perfect image. So our second design was going to try to address that importance of the center of K-space, and we wanted to choose a square spiral trajectory, much like the homework assignment we had earlier. But this one is more dense in the middle, and it gradually gets less and less dense as you go out in K-space and starts to undersample it. This simulates what's most common in modern ramp sampling acquisition trajectories. It's a good way to keep the sampling along the gradients of GX and GY, and the speed of the acquisition process sped up on the outer regions of K-space where the definition isn't, or the information isn't as important to image definition. So as you can see, the new trajectory, the results are much better even without any linear or cubic interpolation. But we still have some missing K-space in the higher frequency values. Those were just filled with 0 on the image on the left. So in order to account for that, we're going to do more compressed sensing. Same parameters as before, just to keep things consistent. We see a little bit higher definition on the left, where the outer regions of K-space are now filled in. You can see overall there's less blurring, and if you zoom in and you see like the fine details and the edges have more contrast, it really results in a better image. The number of iterations I didn't really change, and it didn't really have a dependence on the outcome.
So overall, you can see that the ramp sampling images are not as pretty or with high resolution with the normal acquisition, but they can still produce pretty good images. And this becomes important, like Ying mentioned, for signals that dissolve quickly. So this is the pulse sequence that the original paper that Ying was talking about used, where you can see that they purely have ramps for their GX. Thank you.
So I wasn't really clear on, could you explain a little more about how, so you had an image and then you took the Fourier transform of it, and so you had k-space data that was presumably uniformly sampled.
Yeah.
How did you go from that to non-uniformly sampled?
So I designed the first one that I talked about was giving it like a linear slope, basically just designing a line, and then from that line, because it was just defined by slopes and saying 20% of the points are going to be changing by this much, then I interpolated from the kx value. So basically I made that a line and started taking points, like smaller changes in kx from that line, if that makes sense.
So you created non-uniform sampling.
Yeah.
From the uniform
So yeah, mine was like double interpolated, which is why we also did like more Andrews where we just undersampled k-space.
Okay, great. Was there a question?
Your reconstructions looked really good. Do you have an idea of how extreme the ramp that you used was relative to like what you would see on a real scanner? Did you exaggerate it to show the effect, or is it pretty realistic?
We exaggerated like the first trajectory that you saw, where it's like literally just taking slots out of k-space. You wouldn't really see that in modern imaging. It's going to be more like the second one with the spiral, because it uses like that ramp to accelerate as you go further out in k-space, and it's keeping all of that necessary information in the middle.
Okay, great. Any other questions? So in the interest of time, we'll move on, but there's a question online, so you can just answer that offline. And let's have the next group come up. Next group, oxygen metabolism imaging. Are you in the room?
Yeah.
The next group would be then 5H7, Scarlett. Okay, are you guys presenting about it?
Yeah, is my screen visible? Hello? Are we on mute? What?
Yeah, we are.
No, we are not. Hi, are we on mute? Am I audible?
Exit. Open Zoom.
Expanded. Hello? Hello? Put it in chat, maybe. Oh, we can hear. Oh, okay. It's just time has already started. Sorry for that. Yeah. Hi, everyone. So our project is estimating T1 values using the inversion recovery model and variable flip angle model in MRI. First, what is T1 relaxation time? T1 relaxation time is the longitudinal relaxation time that it takes for the net magnetization vector of any proton to return back to its original state after being perturbed by a radiofrequency pulse. The equation that we have learned in class regarding the magnetization equation and intensity values is given below. Why is T1 important again is that since it is a characteristic of the molecule itself, it can vary from tissue to tissue, thus highlighting a contrast which can be used to image that tissue. T1 values also change with respect to the pathology of the tissue. For example, if there's a disease progressing in the tissue, in case of tumors, the tissue has a changed T1 value, which makes it appear differently in the MRI, thus helping pathological needs. Secondly, how T1 and T2 images are created. This also we have studied in class that for T1 images, we just need to manipulate the gradient echo equation with respect to the TR and TEs to highlight that specific component. For example, if you need a T2 weighted images or T1 weighted images, you can set the TE very, very less as compared to T2, making the equation this, and use it for highlighting only T1 based contrasts. Similarly, you can do the same for T2, highlighting the other part, which changes the contrast. Now, fat in case of T1 has a shorter fat has a shorter T1 value and appears brightly on T1 weighted scans, but appears densely on the T2 weighted scans. And the opposite is true for fluids. Moving on to how we estimate the T1 values. The first model we have is the inversion recovery model. The inversion recovery model was originally designed to null out specific tissues from the images, which is implemented as follows. We provide a 180-degree inverting pulse before the spin echo sequence, which forces all the magnetization vectors to start from the opposite phase and forces it to go through zero while increasing. By carefully selecting the TI value, we can null out a specific tissue and and all the magnetizations beyond that. The results are these for different TI values. How do we use it to estimate T1? First, you collect the data for various inversion times and various images to develop a model between the measured frequency and TI values. Then you use the measured signal intensities for the target image and put it through the model, and using the TI, we can estimate the T1. The next model is the variable flip angle model. The variable flip angle model is comparatively direct. It uses various images taken of the same tissue at different flip angles. So during the same scan, we can apply multiple pulses with different flip angles and collect the data. And for any voxel, the flip angle for which the signal intensity is highest is called the earnest angle. And that earnest angle is dependent directly on T1. So again, how do we use it to map T1? We collect multiple images of the same tissue at different angles, measure the signal intensity, and see which theta it is highest for. But as you can see, since this depends on the argmax of theta, it is highly dependent on the range of theta that we have chosen. So these are the results we have compared further, and Pranati will take over from here.
Thank you, Mayana. So moving on to the methodology, here we are simulating this signal, creating a phantom, and for that we have assumed the values such as T1 to be 800 and TR of IR model to be 2400 and TR of VFA model to be 500. And so this is the configuration that mimics common clinical scenarios. Typically in MRI sequences, TR is greater than T1, which ensures that there is sufficient time for magnetization recovery before the next pulse. And in IR model, we expect the complete magnetization recovery, and hence the value is high, which is 2400, what we have considered here. And when we come to VFA model, the T1 is usually set shorter so that it ensures that the steady state is reached quickly. And once we have simulated the signal using IR model and VFA model, we have added the Gaussian noise to mimic the real data noise, which is usually appears in the real data, something like 2% and 5% and 10% noise. And for the IR model, as Mayana explained, we are varying TI from 50 to 2000. And for VFA model, we are flipping the angles from 5 degrees to 90 degrees. And once we have the signals, simulated signals, we are fitting into a model where we are getting a curve and from which we are estimating the T1 values. And for fitting the curve, we are using least square curve fit function, which is available in MATLAB. And once we have estimated the T1 values, we are also calculating the residual differences. That is nothing but the difference between the estimated T1 values and the actual T1 values, which we have assumed here to be is 800. And then we have visualized the plots. So moving on to the plot here, the noise is increasing as we move from top to bottom. And we can see that as noise increases, the estimated T1 values is diverging. And once we move from left to right,
where the sampling points are increasing, we can see that the estimated T1 values is converging, that is, it is improving as the sampling points increases. And for the same thing, we have visualized the residual plots here. As we can see here, once the noise increases, that we move from top to bottom, the standard deviation is varying largely. But once we move from left to right, where the sampling points are increasing, the standard deviation is almost the same.
Yeah, moving on to the VFA model, compared to the IR model, we can see that the points are more scattered here, unlike IR model where they were quite close to the curve that we were able to fit. So the first point is basically when we increase the noise, here we can see the divergence of T1 around the truth value, which was around 800, is very high in the VFA model. Like when the noise is like around 10%, our estimated T1 value is quite low compared to the truth value. And when we increase the number of sampling points, then our T1 value is improving, is getting closer to the truth value. So these are the residual plots, which basically indicates the error points from the estimated value and the actual value. So here also, like as we move towards higher noise value, the standard deviation of this plots is higher. And when we move from a lesser number of sampling points to higher number of sampling points within the flip angle range that we have set, then the standard deviation is actually pretty much the same. But when it comes to comparing this with that of the IR model, we can see the standard deviation is slightly higher. And so the deviations are quite more, which again proves back the point that here the points are more scattered with respect to the curve compared to the IR model. So here we have just collated the estimated T1 value results for both the IR model and the VFA model for different sampling points for the range and the noise levels. So for the IR model, we can pretty much see that the estimated T1 value is quite around the truth value, which is about 800. And initially, when we tried out with the VFA model, we had chosen the flip angle range to be somewhere between 5 to 60. And as we sampled it, we saw a very high deviation from the truth value, like for example, this case and where it is 640 and another case where it is 466. Then we increased our flip angle range from 5 to 90, where we could see the diversion was slightly lesser, but still worse when compared to the inverse recovery model. So to summarize, basically, we have across these vectors, basically like we have the comparison between the IR and flip angle model. In terms of T1 estimation, we see that generally the IR model is quite more accurate than the flip angle model. And with respect to the number of sampling points that we have, like in case of IR model, different TI values, the sampling, even though we consider lesser number of samples, the IR model gives a slightly more accurate T1 estimation values. But whereas in case of flip angle model, it's highly sensitive to the number of sample points that we take and the range of the flip angles that we actually try out. And then when it comes to noise, the IR model seems to be less prone to noise. But whereas the flip angle model, we can see like if the noise gets added, there was a very high deviation from the estimated T1 value from the truth value. And when it comes to the curve fitting, here in the IR model, it's slightly accurate to fit the curve. But in case of flip angle model, since the truth value that we derived from the model had quite scattered values, so it's slightly harder to actually fit the curve that would probably like have satisfy all those points. So that's why we see more deviation. So yeah, and here are the references that we have. Yeah. Any questions?
Thank you. So can the next group come up since this group is totally remote? Any questions from the room? Yeah. So you might have to ask for the microphone, but what was the
relative scan time of the IR fitting versus the flip angle fitting? Did they take that into account in normalizing?
Yeah, so the question we have from Professor McVeigh is, what was the relative scan time of the IR versus the variable flip angle? And did you take that into account in terms of comparing the two methods?
That aspect we have not compared, but what we have seen is basically like from our offline study that in case of flip angle models, the TR time that is kept is quite less than the T1 value. And it is quite faster than the IR model that we have actually. But the time aspect of this, we have not taken into account in our simulations.
Right. So if you took the same amount of time, you might find out the variable flip angle, you would have many more points then, right?
Yeah.
Okay.
Yeah,
Great.
right.
All right. Very nice. Thank you. Okay.
Thank you.
Okay. Great. You guys are ready. Okay. So
there we go. Sorry about that. Hello, everyone. I'm Shayra with my group mates Skylar and Zoe, and we are going to be presenting the use of echoplanar imaging sequence in diffusion MRI. So first of all, I'll just give kind of a brief overview of diffusion MRI. So it basically is based on measuring the random Brownian motion of water molecules / protons within a voxel of tissue. It basically detects how the water molecules diffuse in the tissue, and they're also affected by different microcellular structures. As a result, it gives us insight into the tissue properties because the structures, which like fibers or like extracellular space or like membranes, can affect how the water diffuses in the tissue, and then we get insight into the tissue property that is not available in conventional MRI imaging because they mostly focus on kind of the anatomical structures of macrostructures. And it's also very helpful in tumor characterization and also cerebral ischemia. That's because tumor changes the tissue architecture. As a result, you'll have different ADC values, and sometimes you can also compare the ADC values to like kind of characterize them as malignant or benign, because benign usually has higher ADC values because they have lower cellular density, so they have higher diffusivity. And for cerebral ischemia, kind of similar, they should have lower ADC values because there would be restricted blood flow. And we use a lot of different sequences for the diffusion map generation, two of the top ones being the spin echo planar sequence and the turbo spin echo. So the ECE207A is mostly basically it uses it generates the case-based data per slice based on a single excitation. On the other hand, the turbo spin echo basically acquires multiple lines of case-based data per excitation by using multiple refocusing pulses in a single TR. And the ECE207A is like mostly used for diffusion MRI, maybe functional MRI, and it's fast and easier than turbo spin echo, but turbo spin echo has like better spatial resolution, and it's better in more complex tissues such as tissues inside the bone or adjacent to the bone because they have minimal T2 signal. So before I go into kind of the acquisition process, these are some of the images that are actually from the MRI lecture 8. So these are the two type of kind of materials or diffusions, the isotropic and anisotropic. Isotropic is more simple and it's in like most like fluids and like gel-like solid. And as you can see, in a perfect isotropic situation, you would have a single diffusion coefficient that's the same along the diagonal column, and then you would have 0 values in the off diagonal. And for anisotropic, it cannot be, it's mostly in like most biological tissues, and it cannot be just one singular coefficient. And it is kind of, oops, sorry, I'll increase so that you can see better. By a 3x3 array, also known as the diffusion tensor. As you can see, the diagonal elements are proportional to the diffusion displacement variances along the 3 directions, and then the off diagonal elements are proportional to the correlation of displacement. So the fundamental property is just like the attenuation of the effective T2 relaxation signal, and then it attenuates based on how easily the water molecules are able to diffuse in a region. So the more diffusivity, the less the initial T2 signal will be present. So we can see it in like different tissue structures in the brain, such as the CSF, which will have a better diffusivity. As a result, more attenuation of T2 and it will be darker. Oh, okay, I will be fast. Just going with the acquisition process, we'll have kind of a 90-degree pulse sequence that rotates the longitudinal magnetization into its transverse axis. And then for the diffusion encoding, which you'll see in our simulation later, we use a biphasic or like bipolar gradients pair of gradient, which is actually adapted from our homework 5. And then for the B value, we use this, I believe it's called the Stenzer something Tanner equation to calculate our B values, where we'll have the gyromagnetic ratio, and then we'll also have like the small delta and the big delta. And okay, do you just want to go? Okay, and then we'll also have the ADC map.
So let me quick overview about the limitations. For example, the limitation to the diffusion MRI, probably the imaging acquisition, which can be easily affected by the artificial noises. Then also they have limitation on the spatial resolution, which also we face on our simulation. Then they have angular resolution, which shown with the B high value. Then they're also dependent, have the trigography dependent, which depends on the users. This guy will show our simulation really quick.
Okay, so our simulation overview. What we attempted to do was simulate diffusion of individual protons in a 256 by 256 matrix. So how this worked was I made a 256 by 256 cell array where each entry in the cell array was a matrix. Each entry has two rows. The top row is the spin and the bottom row is the phases. And the length of the array in that cell entry is the number of protons in that voxel. So the first thing we did was taking as inputs our proton density map, diffusion coefficient map, and our gradients. We calculated the B values. So you can see the function right here, which is the gyromagnetic ratio squared times the magnitude of the gradient in Gauss per centimeter squared times these little delta and big delta, which you can see over here on the right graphically. One limitation of our simulation is that we scaled our gradient amplitudes to achieve B values. This is not how it works in real life. Usually you manipulate the timing and amplitude of a pulse sequence to achieve a new B value. We only manipulated amplitude. So for the actual simulation, for each time step, we iterated through each voxel and updated the spins according to the Larmor frequency equals gamma times sum or net magnetic magnetization formula right here. And then we updated the phases using the spins and the change in time, because spin is just angular velocity. Then for every proton, there was a random walk, so the protons could diffuse, and the magnitude of the random walk was dependent on the diffusivity map. Finally, for the reconstruction, before the simulation, the initial signal maps were saved for each B value, and then running through the simulation gave us the end signal maps. The B values were calculated for each map, as was shown before, and then the reconstruction was calculated for each voxel. So the signal, both the initial signal and the final signal, was calculated as the sum of the phasors given by the protons. And then we used this equation right here for each voxel to just solve for D. And that was our diffusion coefficient map. So some assumptions and limitations of our simulation is we have very little noise compared to real MRI scans. So the only factor contributing to noise in our simulation is random diffusion. As opposed to in real life, you have thermal noise, you have inhomogeneous fields, you have motion artifacts, distortion, all kinds of things that can cause noise that just are not in our simulation at all. Next is our gradients were scaled by a factor of around 2000 to accentuate diffusion effects. This is from the, well, this is not reflecting real life at all. This was only so we could see the magnitude of the change better and better map diffusion. But this was from the block equations where the signal is proportional to the gyromagnetic ratio times the net magnetization. So the net magnetization was calculated at each voxel as B0 plus the gradient at that voxel, Gx at that voxel plus Gy at that voxel. So as you make the gradients larger, they'll have a larger change per unit space, and you'll be able to, it's basically multiplying your diffusion values by a scalar. Our model was only isotropic diffusion, which is actually fine because we're reconstructing apparent diffusion coefficients, which are isotropic anyway. Another assumption of this model was no diffusion across tissue boundaries. So depending on what tissues you're simulating in real life, this could be a more or less accurate assumption. We also had no T1, T2, or T2 star decay, so we had no signal decay over time. The final limitation of our simulation is that it takes forever. As you might imagine, with 1,000 protons per voxel and 256 by 256 voxels, it takes a very long time to run. It took about 12 hours to run on my computer, and I have a very nice desktop. So for our inputs, here is our proton density map. On the left, it's pretty simple, just a Shep-Logan phantom. And then on the right is a diffusion coefficient map I created so that we could see if we could separate the properties of diffusivity from proton density. So, and Zoe made the gradients.
Yeah, so the gradients, we basically use the make API function from the Hallmark 5. So compared to the Hallmark 5, we actually do some modification on the parameters in order to get our desired time points and change the readout time for the DT. And also for the Gx right there, it is our diffusion gradient. And Gy, it actually does not work for the B value, which we kind of not sure, but from my understanding, the Gy is kind of phase encoding gradient. So those two gradients just combine each other to show how the spins in the imaging object, which is the phantoms, are manipulated during the imaging process for each voxel. Like Skylar said before, our phase, so we modified this gradients in order to meet our desired B values.
So as you can see, this is the input proton density map from before the simulation, and then 3 output proton density maps from after the simulation. You can see that the proton density is unchanged within tissues that have individual B values, but in this large middle region, you can see these white circles are present on the input and not on the output. This is because non-constrained portions have diffused to be evenly distributed, which is exactly what we would expect to happen in real life as well. So from the diffusion coefficient map compared to our reconstruction, this is what it looks like. As you can see, as you can hopefully see, not really, but I have a better picture on the next slide. The dots are present here, here, and here, and the lines are present through the image. However, something is wrong with our reconstruction where the B values don't actually weight diffusion. And as you can see, there is some kind of phase shift going on along the y-axis in each of these images. It seems to be slightly shifted based on B value. And if I had to guess, I would say it's from uneven contributions of above and below the curve on the Gy gradient that could maybe cause this. However, I checked the Gy gradient and they had exactly the same area under curve above and below. So that should not happen. This is a blown up image of the previous results where hopefully you guys can see the dots. I know it's kind of hard to see up there, and you can definitely see the line. How do I zoom in, Shire? Go. There you go. And this is the dot right here. I don't know if you guys can see my cursor. It very much, right here. All right. And that is it. Any questions?
Any questions from the room? Yes, go ahead.
So that was really cool. I was curious. I couldn't tell in your gradient plots if you included like a delay between your dephase and rephase gradients. Yeah, so that's kind of my question. Did you include that in your? Okay.
No.
Yeah, because I was wondering, if that could have ended up affecting your final result.
Yeah, I was thinking about that as well. Yeah, because of some edge case that might have caused that GY fluctuation. I was thinking about exactly the same thing, but I checked through and I didn't see the problem, unfortunately. Yeah.
Okay, let's have the next group come up. Great.
Yeah. Hello, everyone. Today, our team will be presenting our implementation and evaluation of deep learning-based interpolation for MRI case-based reconstruction. We'll be focusing on the Raki method. I'm Gokul. I'm joined by Tony and Corey over here to walk through our project. So before we dive in, let me give you a quick overview of what we'll be talking today. So first, we'll start with an introduction to the project and defining the problems we are trying to address. Next, we'll introduce the Raki method and compare its results with GRAPPA for different acceleration rates. Following that, we'll be discussing how Raki's convolution layers and nonlinearity play a role in improving the construction accuracy. Yeah, MRI is a critical imaging tool, but it may take anywhere between 15 to 90 minutes for the reconstruction and like the total one, depending on the target area. So we often undersample the data with multiple coils and then stitch the images together at the final. So some of the methods we learned were like Sensei and Grappa. So for Sensei, it combines the coil sensitivity profiles in the image domain to reconstruct the image, while Grappa uses information from fully sampled calibration regions to develop weighted kernels, which should be applied on the different undersampled regions. So Grappa has some key limitations. So one of the main things is it requires a fully sampled calibration region in the case space known as the auto calibration signal to learn the different kernel weights and also to apply it further. So this reduces the acceleration which is achievable and limiting its efficiency. One more problem is the noise amplification. Grappa struggles with noise. So on the below, we could see some images for acceleration 1 and with a noisy image, and even for higher acceleration rates. So we could see that the Grappa is unable to reconstruct the image cleanly, and also the G factors are deviating away from the ideal value of 1. So what Grappa basically does is in order to evaluate the missing data, it takes into account the acquired data and then the weighted and does a linear combination with the weighted kernels. So these are calculated with the ACS data, which is like fully sampled. So in order to calculate these weighted kernels, it uses the least square method. So if any of the calibration matrix or the target vector is noisy, then the weighted kernels are going to provide a noisy reconstruction. So in class, we have learned about another method called compressed sensing, but the performance is it's very time consuming and we want to improve our time efficiency and accuracy. So we switch from linear mapping to non-linear mapping. So in linear mapping, we know that, for example, y equal to mx plus b, it heavily constructs the data which can be reconstructed and also modeled in that way. So with the same degrees of freedom or even lesser degrees of freedom, the nonlinear mapping reconstructed better, and also it is useful for to adapt to the complexity of MRI data. So applying this nonlinear mapping in MRI reconstruction, we are using CNNs for the convolution layers, and apart from the convolution layers, we also have nonlinear activations like ReLU to adapt to the complex relationships in case-based data.
Now, let me introduce a nonlinear mapping method for MRI reconstructions called Raki. So Raki replaced the linear function GJM in Grappa equation with FJM nonlinear functions. So essentially, Rocky used CNN to approximate this nonlinear function FJM. The network structure is consisted of 3 layers. The input is the originally case-based data. The first layer, F1. F1 applies convolutional filter W1 followed by a nonlinear activation function ReLU. The second layer operates similarly. Finally, the third layer, F3, generates the reconstructed output by applying convolutional filter W3. This is where the reconstructed case-based data is produced. And then here we present the reconstructed image using both Rocky and Grappa with the acceleration factor R set to 2 for both methods. Visually, we can observe that the results are nearly identical. Both methods perform exceptionally well in this scenario. So now let's look at the metrics. The PSNR for Rocky is slightly better than Grappa's. This indicates that for low R, Grappa and Rocky produce similar results. This raises important questions. Why doesn't the nonlinear method Rocky significantly outperform Grappa in this case? Here's the explanations. At low R, the reconstruction problem is similar because a relative large portion of case-based data is retrained. In such cases, Grappa's linear assumptions are sufficient for good performance. However, Rocky's ability to regularize better and capture subtle nonlinearity gives us a slightly better result. Since we've established that both methods perform well at low R, so next question is, what happened when we increase R? This is a result for Grappa and Rocky at R increased from 2 to 6. A higher R, like R from 5 or 6, Rocky demonstrates visibly improved noise properties, whereas Grappa suffers from significant noise amplifications. Let's take a closer look at the reconstruction at R equals to 6. When we zoom in on both images, we find the first arrow is the horizontal line artifacts, and the second one is the severe background aliasing. So in contrast, Rocky produced much cleaner images, maintaining decent reconstruction quality even at the high R. So why does this happen? To understand why Rocky outperforms Grappa at high R, let's examine the case-based data. In this diagram for low R, a blue region indicates the ACS area. Grappa leveraged this ACS region to linear estimate the relationship between neighboring points. The calculated weight W are then applied to the undersample case-based data to interpolate the missing lines. However, with high R, more data is missing, creating larger gaps in case-based. The increased variability in data outside the ACS regions makes the ACS less representative for the entire case-based. Grappa fixed shift invariant weights fails to generate well to these larger gaps, leading to artifacts and aliasing in the reconstructed image. On the other hand, Rocky used CNN to model the relationship between neighboring case-based points and the missing data lines. Unlike Grappa's linear weights, Rocky can approximate complex nonlinear dependency, which are essential for addressing larger gaps in case-based. The Rocky tailored weight learning by training a neural network directly on ACS data, which is here in the blue regions, learning weights that are specific to the call sensitivity, spatial correlations, and sampling patterns of the data set, allowing Rocky to handle the challenge of a high R more effectively.
Okay, now we want to explain why Rocky is better than Grappa, although both of them have kernel operation. First, Grappa assumes the local feature in case-based are consistent, while Rocky uses the weighted kernel to extract local variation. And in this case, the backpropagation of CNN allows each weight to get feedback in 2 dimensions. And secondly, Rocky applies nonlinear estimation to approximate complex pattern of case-based by the ReLU function. With such information, Rocky can apply on smaller ACS regions. In other words, it is more robust than Grappa. As a result, we can conclude that Grappa might propagate or amplify the noise, and then leads to some problems like aliasing or oversmoothing. Okay, to support our statement, we do some experiments. And for each experiment, we retrain the model actually to get a better performance. Okay, in this slide, we want to discuss how the ReLU function influences the Rocky model and to see if it downgrades to Grappa. After we remove the ReLU from the Rocky, you can see that some noise appears in the reconstructed image. As part of our experiment, we also remove only the first ReLU or the second ReLU function. And we also found that the model performs well when only removing one ReLU function. Yeah. And while removing all the ReLU function leads to worse results. And from our opinions, the sequence of ReLU
is important because the early stage ReLU leads to lower PSNR. And why ReLU is so important for Rocky? Without ReLU, when the model operates the backpropagation, the zero value region in the subsample K space will contribute gradients to the loss function because the linear function has constant loss. It's just like the level 1 regularization, that the gradient is always a constant. Okay, and hence those supposed inactive regions interrupt the information we want. But there's one interesting here. Since Rocky still have the weighty kernel convolution, it's still better than the nearest neighbor mechanism in Grappa for searching some useful information in K space. Okay, next we are going to talk about the comparison of Rocky and Grappa when we're removing some convolutional layers. We had done 2 experiments. One was removing the whole layer, and the other was 2 layers. Yeah, and from the result, we can see that we get higher PSNR if we remove only one convolutional layer. For this phenomenon, we point out some potential reasons. The first one is the most straightforward one. With a deeper network, the model can construct a more complex function, just like higher degree of polynomial in linear regression. And secondly, a deeper CNN model will lead to a wider receptive field for the model to check the K-space more globally. Okay, in conclusion, we dive into the comparison between Grappa and Rocky for NRI K-space reconstruction. First of all, we found out that the Grappa has limited expressiveness so that it leads to more noises and it cannot adapt to more complex K-space patterns. And then we found that Rocky includes nonlinearity and gets better performance. And finally, we did experiments like comparing Rocky and Grappa on the different assertion rates, like 2 and 6. And we also removed the ReLU function and changed the convolutional layer of Rocky. And based on our exploration, we found that nonlinearity and shear weighting in the kernel are really important for good reconstructed NRI image. That's our project. Thank you.
Oh yeah, Professor McVeigh, go ahead.
It wasn't clear to me, or maybe I just missed it. The Rocky training, but was there a ton of training already done to make Rocky versus this one thing you get out of Grappa, which is the central?
Sorry, so your question is that there's a lot of trainings in Rocky.
Right.
And
does it come to the party with a lot of previous images and training on board, right?
Right.
Yeah. Okay. But how much is that in the Rocky that you guys downloaded? Is all that training's been done?
Yeah, the training in Rocky, it does contains a lot of images in the data sets.
Okay.
Yeah.
But you guys retrained it.
Yeah, we did retrain it. So we have like different results. So we can remove the layer. So after we remove each layer, we do retrain it. So yeah.
Can you go back to yeah, this one. You said you removed.
So originally, please look at the flowchart under the title. So originally the layer sequence will become like this. This is the most left images, so Rocky R equals to 6. And then we remove this one, convolutional plus the ReLU, which represents the second layer. So the result will be the middle part. And then we remove all of the first 2 layers, and the result will be the last one. So the whole goal is that we explain that the greater network depth enhance the weight sharing because there's more CNN stacking together.
So is it because the I mean the last convolutional layer could just be a Grappa-like kernel? So why doesn't it do as well as Grappa?
Because I think in our opinion, the convolution filters still have to some extent have some better perform than Rocky, and we noticed that because their kernel size are different.
Oh, so yeah, the question is, so your last layer that you kept, was that kernel size smaller than the Grappa?
Yes.
It was.
Yes. So the kernel size for Grappa is 5 times 5, and then for this one is 3 times 3. So this also raises another pros for Rocky because it requires less kernel size. So it requires less ACS regions. So they don't need a lot of fully sampled ACS regions. Thank you.
Thank you. make sure you're on. Okay. Yeah.
Okay, hello everyone. I'm Devanshi, and these are my teammates Achyut and Ishita. And today we'll be presenting our project on comparison and implementation of sense and grappa reconstruction algorithms in parallel MRI. So, as we know, traditionally MRI acquires data sequentially, which can be very time consuming. So, parallel imaging aims to tackle that challenge by speeding up the process while maintaining the image quality. So, parallel imaging involves using multiple coils with each with a unique sensitivity profile to gather the data. You then undersample the case space and then using advanced reconstruction techniques that are based on spatial encoding, you construct a a full FOV image of the original data. So today we'll be discussing two techniques, sense and grappa. The first technique that we implemented was sense, which operates in the image domain, directly reconstructing the image from the undersampled data using the coil sensitivity maps. So the coil sensitivity maps actually help us identify how each coil sees a specific part of the same image. So here, if you see the points A and B in the original image, in the undersampled image, they actually get combined because of aliasing. But since we have the measured coil sensitivities at points A and B in the form of our sensitivity matrix, and we also have the measured alias pixel values P1 and P2 from both the coils coil 1 and coil 2 at these points, we can get the true signals at point A and B by simultaneously solving these equations. So, which helps us construct the full FOV image. The next technique we implemented was grappa. So that is a parallel imaging technique that reconstructs the undersampled MRI data in the K-space domain. So here, if you see this kernel, each of the different colors signifies a different coil. So, and this black box over here signifies the K-space that we'll be sampling. This K-space is collected over each of these coils. And we also collect an additional data in the form of the auto-calibration signals or ACS, which is then used to calculate the grappa weights by collecting the kernel repetitions over the ACS region. These grappa weights are then applied to our undersampled K-space data to generate the missing K-space data points for each of the coils. Each of these coils are then we then apply Fourier transform to each of these coils to generate a single coil image, and then which is then combined to get a full FOV image. The key difference between sense and grappa would be that sense is based on the image on undersampled image regeneration, but a grappa is based on a K-space regeneration, which also gets us to a next key difference that grappa performs better in complex and non-uniform sensitivity regions, whereas sense performs better when the coil profiles are more uniform. Our simulation setup in MATLAB was that we used this original image from a brain MRI scan. We also simulated it for two coil profiles, Gaussian and sigmoid, and we varied the number of coils from 4, 8 to 16. We also used R factors 1, 2, 4, 6, and 8, and our evaluation metrics were mainly focused on MSE and SNR.
So I'll be explaining the flowchart here that we have for sense and grappa. Initially, we see that we start by producing the coil profiles, and then we calculate the Fourier transform and then perform an inverse Fourier transform so that we get the individual like coil images. And then after that, we estimate the coil sensitivities. This basically helps focus on different parts of the image through each of the coils. After that, we smooth and mask the sensitivity map so that we enhance the accuracy and reduce noise. Then we produce the optimal combined image using the estimated coil sensitivities. We then undersample the K-space using different R factors so that we can accelerate the imaging process. Then we finally reconstruct the final image where we solve the final inverse equation. For grappa, we again we produce the coil profiles and then calculate the Fourier transform. We then undersample the K-space using different R factors so that we can accelerate the imaging process. We then estimate the weights based on the calibration data, and then we fill in the missing gaps using interpolation and then nearest neighbor data. And then after that, we perform inverse Fourier transform across each of the coils to get the individual coil profiles. Then we reconstruct the final image using the RMSE square method, where we combine that across all the individual images that were generated. This is the slide that shows how, with changing the acceleration factor, how the reconstruction works for both sense and grappa. We see that as the R value increases, the reconstruction is getting much worse. We have chosen the Gaussian profile here, and the number of coils is fixed as to be 16. And we see that for an R value of 2, the grappa reconstruction almost resembles the original image, but there are a lot of streaks that are observed in the sense reconstruction. For an R value of 8, we see that the sense reconstruction is a lot worse. There's a lot of aliasing that we observe. But the grappa reconstruction, we see that it's a little bit hazy, but still it performs much better than sense reconstruction for this high undersampling scenario. So these are the different plots that we got. So for this, we have used the modified Shep-Logan image. The reason being that there was no input noise that was occurring with this. The image that we chose that had a little noise, due to which the MSR and SNR values weren't indicative of the sense and grappa values that we wanted to estimate for that. So here we see that as the R value increases, the MSE increases and the SNR decreases, which is expected as the image quality is getting a lot worse. We also have some key observations, being that Gaussian coil profile is doing much better than the sigmoid coil profile, as we can see with the MSE and the SNR values that are present. And one more key observation is that for high undersampling values, grappa is performing much better than the sense, due to, as we can see from the MSE values that are present here. We also vary the number of coils and then see how grappa and sense perform for reconstruction. We see that for sigmoid coil profile and for R value being fixed as 2, as the number of coils increases, the reconstruction is getting better. We see that for the number of coils has to be 16, grappa reconstruction almost resembles the original image, but there's some streaks present in the sense reconstruction, which shows that grappa is a much better method for reconstruction compared to sense. And in this plot, we reinforce the idea that as we increase the number of coils, the MSE is decreasing, whereas this SNR is increasing. And this is expected as the image quality is getting better with number of coils. We also see that Gaussian profile is doing much better than the sigmoid profile, as it is evident to the SNR and MSE values here as well.
So now, taking over for the coil profile variation, we chose Gaussian and sigmoid. The coil sensitivity profiles usually detect the signal intensities across the field of view, and we can see that the coil sensitivity profiles match the shape of the function chosen. Like for Gaussian, it's stronger in the center and then tapers off towards the end, while for sigmoid, it's a bit flatter in the center and then a little steeper at the boundaries. And this is the grappa and sense reconstruction using Gaussian and sigmoid for R2 and number of coils 8. We can see that for Gaussian, grappa and sense both performed a bit better, and this is due to Gaussian having a strong localization and minimum overlapping. Whereas for sigmoid, we can see grappa is a little hazy because of its broader coverage, and sense has a lot of aliasing, once again reinforcing that Gaussian performed much better than sigmoid. We also wanted to do another additional experiment where we use sinusoidal coil sensitivity profiles because they have periodic oscillations in their sensitivity. And we can see that there was a lot of image degradation for it. In grappa and sense, we can see a lot of aliasing, repetitions, and streaks. So since Gaussian and sigmoid perform much better, we stuck with them for all of our analysis and results. So earlier we were seeing all of the results with keeping one variable constant, but here we can see all of the R and all of the coil values. The rows indicate the number of coils 4, 8, and 16, and the columns indicate R2, 4, 6, and 8. And we can see that as R increases, the image quality is degrading. There are a lot of artifacts, but as we increase the number of coils, the image quality gets better. There are structural details which are preserved, and we can see that even for a higher R like 8, for increasing the number of coils, we get a pretty decent image. And this is for grappa sigmoid, that we can see that there's a similar trend where as R increases, the image quality degrades, and as the number of coils increases, the image quality gets better. But it does perform a little worse compared to Gaussian if you compare. And this is because for grappa, Gaussian has like a smoothing kernel whereas like sigmoid has a steeping kernel, which is not as robust as Gaussian. We see a similar trend for sense Gaussian as well, but if you look for R8 coil 16, the image quality is pretty bad, which sort of shows the limitations of sense as compared to grappa. And when we come to sense sigmoid, the image for R8 coil 16 or even the other higher R8s is pretty much unusable, which again shows the limitations of sigmoid over Gaussian and sense over grappa. So we want to sort of conclude everything. So we set out to measure sense in grappa using metrics like MSC and SNR. And what we observed is that as R increases, the undersampling is more and more artifacts are seen. And hence we saw that the SNR decreases and the MSC increases. But for grappa, it was a little more consistent. And as the number of coils increased, the MSC decreased, whereas the SNR increased. So we were able to get a faster image, and we did not compromise on the image quality, but it was computationally more intensive. And coming to the artifacts for sense, we saw a little more streaking or ghosting artifacts, whereas for grappa, it was a bit more hazy appearance. And this is due to like grappa's limitation in interpolation when undersampling, and for sense, that's due to the sensitivity coil inaccuracies. And for both sense and grappa, Gaussian performed better than sigmoid and much better than sinusoidal. So when we want to choose our technique, we have to take into account what parameters we want, what coil profiles we want, and also take into account the practical considerations like the timing, the reconstruction efficiency, or like the cost effectiveness, so that we can strike a good balance and leverage all the benefits we want. And these were our references, and any questions if you have.
So for 16 coils, you just have them circulate around the brain? Like, what was the positioning of 16 coils?
So we
What was the layout?
randomize that. So it randomly generates the profiles, and then it's very different compared to whatever is the coil profiles that are present. So it chooses different parts from that.
Okay, so like, I mean, but can you describe what the coil layout looked like for 16 coils? Like,
Oh.
what did it look like physically?
Oh, so for the 16th, so given a coil profile, so it shows like different image, like one image, like the coil profile was here, and other it focused on different part of the brain. Then for other, it like chose on different part of the brain. So it covered mostly all the regions that were present, and then we sort of chose that for then reconstructing.
And so
that.
did you, I mean, I'm surprised the sense did so poorly. So did you try with R equals 1 as a sanity check?
Yeah, we have did with R equals 1. We haven't put in here, but
How did that look?
I think for R equals to 1.
It was almost like the original image.
Yeah.
But like little artifacts were seen. Very little. Yeah, it was pretty much like the original, but little artifacts I think we saw.
Oh, R equals to 2.
Yeah, there's sense something happened with sense that it got, there's a bug in there.
Yeah, there's something wrong with your encoding matrix probably, because there shouldn't be that streak there.
Yeah, the
Oh,
streak
okay.
is not, sense at rate 2 or 4 coils, it should just look like the original.
Okay.
Okay.
Yeah. But
Maybe
otherwise,
that
really,
could be how we
it's-
coded it.
I mean, you guys put a lot of effort into it, so
Yeah.
it's fine that there's a bug, but there probably is a bug.
Oh.
Yeah, it's,
Okay.
but it's a great, it's a really comprehensive simulation.
Yeah, no, very good. Okay.
You can move it around and stuff, but
Okay.
you tweak it, you should be able to get that sense to work.
Yeah, I think probably one reason might be the input image was sort of little hazy. Probably if we chose a different input image and we
No,
tried that out.
there's just a bug in your code.
Oh, okay. We'll try to look into that then. Thank you for pointing it out.
Any other questions? Okay, great, thank you.
Thank you.
Yeah, next group, come on up and share your screen. Go ahead.
Hello everyone. So this presentation is about motion correction in fMRI acquisition. We first discuss a little bit about fMRI data acquisition first. And I'm Jason, this is Ella, and this is Eric. Okay. So let's start with both signals. So neural activity increases the concentration of oxygenated hemoglobin relative to deoxygenated ones. And deoxygenated hemoglobin is paramagnetic, while oxygenated hemoglobin is not. So the difference, so this difference affects the local magnetic environment, result in both signal changes. And fMRI measure those change, so it detects both signals from protons. And in regions with higher levels of deoxygenated hemoglobin, the local magnetic field becomes less uniform. That increases T2 star relaxation and in the nearby hydrogen protons and leading to a weaker Bose signal. And we have to use fast imaging sequences like EPI to capture these Bose signals. We've discussed why later. And as we can see in this picture over here, so wait, we can see. For some reason, the color is reversed
Yeah,
here.
don't worry about it. That's-
Yeah. So actually, what you are looking over there is red, but it's actually blue. So the blue region shows a decrease in the both signal, and the red region over here shows an increase of both signals. That means the brain activity is actually higher here compared to here. Okay, and the data acquisition of fMRI is a little bit different from normal MRI. So in fMRI, we track changes in both signal over time. So the image is not static. And for that reason, the temporal resolution is prioritized. And that's the reason why we need fast imaging techniques like EPI over here. And we also have to use gradient echo instead of spin echo because gradient echo is sensitive to T2 star relaxation. And fMRI is heavily depends on T2 star relaxation. So what is T2 star relaxation? And as we all know, T2 describes the natural decay of transverse magnetization due to spin-spin interactions. And T2 star is similar to that, but it describes the effective transverse relaxation time, accounting to T2 decay and additional dephasing caused by local magnetic field inhomogeneities. And which means this little local magnetic field inhomogeneity is caused by the oxygenation of the hemoglobins. And generally in fMRI, we don't like any motion effect because the sharpness of the image is reduced because of motion, and motion artifact can be observed. And as we can see in this picture over here, the top picture has motion simulated, and the bottom picture is without. So we can see there are a little bit stripes over here and here. So they are definitely interfering our observation of the images. And fMRI is particularly sensitive to this kind of motion because it causes voxel misalignment, corrupting the time series data. So basically, we want to monitor the brain activity in a region, but with motion, our data may actually come from another region. So we don't want that. There are multiple strategies like to deal with this problem, like shorter scan time, patient restraint, and we are gonna talk about motion correction algorithms next. So I'm gonna introduce a technique that is crucial for improving the quality of MRI data. It's the prospective motion correction, which is PMC. As we know, motion during fMRI scans can be significantly affect image quality, leading to inaccurate result. PMC addresses this by detecting motion in real time and adjusting the scan accordingly. So let's dive into the first aspect of PMC, motion detection and parameter calculation. During fMRI scanning, hand motion is head motion is detected using one of three main techniques: optical tracking, navigator techniques, and field detection method. During fMRI scanning process, the system use optical tracking, magnetic field detection, or navigator techniques to detect head motion. So the optical methods, the optical tracking system are employed to monitor head movement by using cameras. These cameras track external markers placed on the subject's head by analyzing the position of orientation of these markers in 3D space. The system can calculate head movement in real time. This allows for quick adjustment to be made during scanning. So as you can see, figure A is the camera placement. An MRI-compatible camera is positioned between the transmit-receive portion of the head coil. This setup allows for continuous tracking of head motion during the scan. Figure B is the optical markers. It shows the checkerboard optical marker placed on the subject's forehead. This pattern provides precise reference points for camera to track motion. Figure C is the rigid tracking. It demonstrates the marker's secure placement on the forehead for accurate rigid tracking for head movement. The second method is the navigator techniques. Navigator techniques utilize the MRI scanner itself to measure motion. During the scanning process, additional small MR signals known as navigator echoes are acquired. These echoes help in detecting motion by analyzing shifts in the phase of frequency of the MR data, providing crucial information about any changes in the subject's position. The next method is the field detection method. Small magnetic field sensors are another method used for motion detection. These sensors often integrate with RF coils or as separate device. These are attached to subject's head and they detect changes in the local magnetic field as the subject moves, allowing for precise calculation of both rotational R of t and translation T of t. By utilizing these three methods, we can efficiently detect the calculation motion during the fMRI process. This data is essential for the next step, real-time adjustment for gradient waveforms.
So in fMRI, motion is typically modeled as rigid body movement, a combination of rotation and translations. And that starts with the rigid body motion equation. Here, X is the original position of a point in the field of view, and R of t is the rotation matrix describing how the object rotates over time. T of t is the translation vector representing how the object moves linearly. And X prime of t is the new position of the point after motion. So this equation is the foundation of modeling motion in MRI. And in fMRI, the object such as a brain slice is represented as a grid of points in 3D space. These points are stored as a 3 times n matrix, where each column represents a point in the grid. And now let's focus on the rotation matrix R of t. It is a 3 times 3 matrix that describes how the object rotates around the x and y and z axis. Each individual rotation is represented as a rotation matrix, as it shows. And 3 rotation matrices will then be combined together. Next is the translation vector T of t. It represents the linear motion of the object along x, y, and z axis. And then we can combine rotation and translation to calculate the new position of all points in the f of v. And this process can be applied to all points in the f of v simultaneously. So the next step is to update magnetic field gradient waveforms. To compensate for motion, the gradient waveform g of t must be updated during the process when motion is detected. And the original gradient waveform g of t is transformed by the rotation matrix. The updated gradient waveform ensures that spatial encoding remains accurate calculate this by motion. The corrected MRI signal is given by this equation, where S of K and T is the original signal in K space, and delta phi of T is the phase shift caused by motion. Last, S prime of K and T represents the corrected signal. This phase shift is derived from the motion parameters R of T and T of T, and this ensures spatial alignment for accurate image reconstruction. And this is the result of PMC research. Both figure B and D are images with motion, and figure D has undergone PMC. And we can see that PMC have significant success, while figure B has severe ripple effects, and figure D shows a clear image. In a nutshell, prospective motion correction is a powerful real-time method to address motion artifacts in MRI, ensuring high-quality imaging even with patient movement. Thank you. And this is our reference.
Okay, so are there any downsides to using prospective motion correction?
Downside. So I think the most problem is the time, how fast it can update the gradient waveform. Yeah, and I see most of the paper, the fastest is about 200 millisecond right now. Yeah, so if it is better to speed up to under 100 millisecond. Yeah, so I think this is the most problem right now.
Okay, great. Thank you.
Thank you.
Thank you.
Hey Dante, can everybody hear me?
Okay, can you hear me?
Yeah, I can hear you. Can everybody else in the room hear me though?
Josh, you ready?
Yeah.
Yeah, Joshua, yeah.
Yeah.
Everybody can hear me? Okay, cool.
Yeah, great.
All right. Good afternoon, everyone. I'm Josh here with Dante and Steven, and our project focuses on implementing deterministic streamlined tractography through open source and MATLAB toolboxes to visualize brain graphs. Next slide, please.
Yep. Ooh, sorry, hold on.
Okay.
It went away. So yeah, sorry, technical difficulties. Okay, there we go.
Oh, one back.
There we go.
Okay, there you go. So diffusion tensor imaging, or DTI, is foundationally based upon the anisotropic diffusion of water molecules. So in the brain, this directional movement is dictated by the microstructure of tissues. By leveraging these diffusion patterns, DTI enables us to visualize and investigate the brain structural connectivity. So as you can see here, the images on the slide illustrate that without diffusion weighting, structural details are unclear. But with diffusion weighting, you can discern detailed microstructural differences, such as axon fiber orientation and density, which are crucial for studying brain function and pathology. So DTI is used to explore neurological conditions like autism spectrum disorder, where visualization of white matter abnormalities help us understand the disease's impact on cognition or behavior. Next slide, please. So DTI, again, relies on introducing dephasing and rephasing gradients, which make MRI sensitive to molecular movement. So the degree of signal attenuation is determined by three factors. That is mean travel distance, phase shifts caused by the gradient, and the population of molecules moved by that gradient. The final signal decay, shown as equation 3.8, shows how the signal is attenuated. Here, S naught is the original signal without diffusion weighting, D is the diffusion constant, and B is a parameter summarizing the gradient factors. To create meaningful diffusion weighted images, at least 6 unique views or B vectors are typically used, though more can improve accuracy. These vectors define the directions along which gradients are applied, allowing us to calculate diffusion constants along multiple axes and capture directional movement of water molecules in the brain. Next slide, please.
Yeah, I'm trying. There's an annotate tool right in my way. Okay.
Oh, I see. Okay. And so by mapping these diffusion patterns, DTI enables tractography, a computational method for reconstructing the white matter fiber pathways. We chose deterministic streamline tractography, DST, along with the Dippy Python package for its simplicity given the tools and the time that we have. So DST is a widely used algorithm that reconstructs white matter fiber tracks by following the principal eigenvector of the diffusion tensor each voxel. Starting from user-defined seed points, the algorithm propagates streamlines iteratively in the direction of maximum diffusion until reaching a threshold. And so our general workflow follows this chart. So the first step is just to load the patient data from OSF into Colab. Step 2 is to denoise raw data using PCA. Third step is to correct subject movement during sampling for image alignment and artifact reduction. Step 4 is to fit the diffusion tensor with the denoise data to determine fractional anisotropy. Step 5, define those seed points to iteratively propagate streamlines to our desired threshold. And the last step is to visualize the generated streamlines to map white matter connectivity. Next slide, please. And also a quick comment on the data set. So the Open Science Framework has raw DMRI data of 80 participants, 40 are female, 40 are male, that were taken on a 3 Tesla Trio Siemens scanner. The DWIs were acquired using spin echo EPI sequences with the parameters listed. And since the file sizes were massive and computation time was long, we decided to only image patient 2 as they have no excluded volumes according to the paper. Next slide, please.
All right, so I'm going to talk a little bit about one of the implementations that we did. So the first implementation was done using Python in a Google Colab notebook. So to implement this, we loaded the data for participant number 2 into the notebook. Because of the time required to process the whole data set, we decided to first reduce its size, and we only include the first 16 volumes. The reduced data set then underwent denoising by PCA, and this was followed by motion correction prior to fitting the diffusion tensor model. You can see the image before and after the denoising on the right-hand side. So our goal in fitting the diffusion tensor is to learn the eigenvectors and eigenvalues at each voxel corresponding to the principal directions of diffusion at that location. To do this, we measured the apparent diffusion coefficients at each location using gradients in different orientations given by the B values and B vectors. This allows us to learn the diffusion tensor, which can be diagonalized to return the eigenvalues and eigenvectors that we're looking for. Once we have the diffusion tensor model fit, we can calculate a measure of diffusion at each voxel. In this case, we calculate the fractional anisotropy given by the equation in the lower left. Before we can generate the streamlines, we have to set a fractional anisotropy threshold to distinguish white and gray matter. And you can see on the top right-hand side, the full fractional anisotropy map, and the bottom one is once we've applied a threshold of 0.2. So we use that to determine our seeds from which we will generate our streamlines. And so having defined those seeds, we use a built-in deterministic tractography algorithm that iteratively constructs streamlines from our seed locations. Streamline generation is terminated upon reaching a stopping criteria, which we have set to be the fractional anisotropy threshold of 0.2. And so specifically for our implementation, we use the diffusion imaging Python or DIPI's local tracking function to calculate streamlines from seed voxels and computed orientations at each location. Once generated, streamlines were saved and visualized in Google Colab as pictured on the right. And so Stephen will take over for the next implementation.
So essentially something we wanted to do was kind of compare the results that we got from the Dippy implementation with some other toolboxes that we were able to find. And one of the toolboxes that we found was the DTI fiber tractography streamline tracking technique toolbox on MATLAB. And this one actually computes the fractional anisotropies, eigenvalues, and the diffusion tensors all out of the box. We just had to adapt it to fit our DWI and the BVEX and BVALs that we pre-processed. So essentially, as we just mentioned, we're comparing the same eddy corrected denoise DWI that we got from pre-processing. And then when we pass this into the MATLAB toolbox, it essentially computes its own FA matrix, which we've essentially displayed a slice, the corresponding slice that we also displayed earlier. And straight off the bat, we can see from the top two images, the colors inverted, that the FA values that we generate from the MATLAB toolbox is actually a bit lower than what we generated. And if we do the same thresholding value, we actually get a mask that also has less streamlined fibers. And this actually kind of changes what the information that we actually get. And essentially, this tells us that this allows us to highlight some of the denser white matter tracks, while also cutting out some of the looser and less dense white matter tracks, as well as the gray matter tracks that might have been included in our original implementation. And then next, the MATLAB toolbox essentially generated a 3D render. And this time we have multiple viewpoints of it. And we've essentially provided the top-down matching our DIPI render, and then the posterior and then the back left views. If we look at the right three images first, we've essentially applied the same FA threshold of 0.2. And as we expected, we do see a lot less streamlines. And if you look at the actual streamlines generated, it actually does also highlight the denser and more organized regions of our brain. And if we compare it with the left side, which is a lower threshold, we see a lot more of those streamlines. And the left side actually matches more of what we generated with DIPI. And essentially, if we compare these two, we can actually see the importance of how thresholds dictate what we actually see in our tractography. If we have a higher threshold, we'll see more of the concentrated, organized white fiber tracks, and also like it tells us more of the major connections between parts of the brain. And in a lower threshold, we'll see the more loosely connected, or the looser and less dense areas, and also the connections on the outer parts of the brain. And so to kind of conclude, we essentially implemented the DST using our open source DWI dataset, and we were able to successfully generate the streamlines. And then the streamline generations can kind of be used to construct structural connectivity matrices, which we can use to analyze with other tools and graph theory and network science to kind of see how our brain activities, and also possibly look into possible brain damage or brain injuries or any other type of diagnosis. And then using our comparisons, we can see that the importance of the threshold, as we mentioned, increasing the threshold isolates more of the white matter tracks, and it also kind of determines the density of the streamlined fibers that we get to see in the tractography. And in our final comparison as well, we do actually see that the clarity of our fiber tracks also matter depending on the toolboxes we use. And then here's our reference. Any questions?
I'll have a question. So did you look into why you got different FA values for the two toolboxes? I mean,
Yeah, so the toolbox we use actually had provided their derivation, and if we were to compare, I don't have the image provided, but if we were to compare their calculation for the eigenvalues versus ours, they actually use the mean diffusion, or the mean eigenvalues, instead of just calculating the difference between the two different like eigenvalues.
okay.
So we're thinking that's probably one of the major causes, since that was I think the major difference between our different derivations.
Okay. Thanks. All right. I think the last group
is presenting remotely because of they're not feeling well. So Are you guys, is the last group here? So this, you guys are group
Yes.
23, right?
Yeah.
Group 24, are you online?
Yeah, we're online.
Yeah, we're here.
Oh, yeah. We're here.
Michael, yes, go ahead and share your screen.
Mhm. All right, hi everyone. We're group 24, and we did a presentation on the effect of the flip angle and other parameters on the time of flight, MR signals, strength, and contrast. My name is Michael.
I'm Viha.
I'm Andrew. So this is our table of contents. And for our introduction, we're gonna go over the time of flight and geography. So the time of flight angiography is a magnetic resonance imaging MRI-based vascular imaging technique, and it's widely used to visualize the blood flow in areas such as cerebral vessel and carotid arteries. And essentially, it's based on the phenomenon of the flow-related signal hyperintensity, and this is dependent on the spins entering into an imaging slice. So TOF imaging is able to exploit signal differences between flowing blood along with stationary tissues to produce high contrast vascular images. And this technique is particularly useful for detecting vascular abnormalities such as aneurysms and stenosis. And for some of the applications and clinical significance for our topic, so for the TOF imaging, we were able to identify the cerebrovascular evaluation. So for instance, detecting aneurysms, malformations, along with vascular occlusions, along with stenosis in the brain. And then we also looked into neck vessels, so being able to assess carotid artery disease, and then spinal cord vascular evaluation. So spinal cord ischemia, along with peripheral vascular disease as well. So that's in the legs, arms, or abdomen. And then for the clinical significance of TOF imaging, we identified it's non-invasive, so it doesn't require the insertion of catheters. It also has high image quality, so it provides detailed anatomical information about the blood vessels, and also early detection as well. So being able to aid in the early detection of vascular pathologies, which allows for timely intervention and treatment planning, along with being able to monitor the treatment efficacy, so monitoring the effectiveness of treatments for vascular conditions. And for the objective of our project, in our project, we aim to focus on one key imaging parameter, and that's the flip angle and its effect of TOF signal strength and contrast. So in our presentation, we analyze equation 15.27 from chapter 15, which is the angiographic pulse sequences from the Canvas homepage. And that's the equation. And essentially, we're going to evaluate S, which is the signal intensity, T1, which is longitudinal relaxation time, T2, which is a transverse relaxation time, TR, which is the repetition time, and then TE echo time, and theta, which is the flip angle, and then M0, which is the equilibrium magnetization.
So talking about how the concepts that we covered in class relate to our project, they relate in three main ways. First is the concept of longitudinal relaxation, also called T1 relaxation, which, as we learned in class, refers to the process by which the net magnetization vector realigns with the external magnetic field. And so this basically determines in TOF imaging how quickly the magnetization vector or how quickly the magnetization recovers along the longitudinal axis. Then another concept which we briefly touched upon in class was flow-related enhancement, which is a phenomena in MRI which leverages the difference between fresh blood and stationary tissue. We will discuss more about this as we dive into the presentation. And it basically produces a contrast, which is key in producing high-quality vascular images. And finally, the role of flip angle. So as we discussed in class, the flip angle refers to an angle by which the magnetic moments of protons are tilted away from the main magnetic field during an RF excitation pulse. As you can see in the image here, alpha is the flip angle, which is tilted away from the main magnetization field, B.
The flip angle determines how much the magnetization vector is rotated by the RF pulse, and in this way, it directly affects the signal strength and contrast. Could you go back a slide?
Yeah.
So the flip angle is a pivotal parameter in TOF-MRA, and this can be studied in two ways. What happens when we increase the flip angle and what happens when we decrease it. So when we increase the flip angle, it increases the signal from the inflowing blood. The way this happens is that the blood which is entering our imaging slice has not yet been exposed to any prior RF pulses. So you can think of it as fresh blood. And a higher flip angle is more likely to convert more of the longitudinal magnetization into transverse magnetization. What we detect as signal is basically this transverse magnetization. So since the blood is fresh, as in it hasn't been exposed to any prior RF pulses, more of it can be detected into signal, and this enhances the visibility of vascular structures which have flowing blood in them. Increasing the flip angle also decreases signal from the static tissue. This is because static tissue was always there in our imaging slice, so it has already been exposed to repeated RF pulses several times, and this leads to its saturation over time, which decreases its main signal and hence produces this contrast between the flowing blood and the static tissue. However, there's also a drawback here, is that a higher flip angle can also result in signal saturation of the flowing blood itself, particularly in places where the blood is not moving as fast or if we're taking thicker imaging slabs. On the other hand, decreasing the flip angle reduces these negative saturation effects on the blood, but it also preserves signal from the stationary tissue, which eventually reduces our contrast between fresh blood and the static tissue. So as you can tell here, it is pivotal to determine an intermediate flip angle in order to balance all these competing factors. And we can do so by taking into account several other parameters, such as blood flow velocity, slice thickness, and repetition time, some of which we will discuss in the presentation.
So for the 2D TOF imaging, it involved acquiring multiple thick slices, typically 1 to 2 millimeter thick, perpendicular to the blood vessels. Making it suitable for imaging long vessels like the aorta or carotides. Its advantages include shorter imaging time and expandable coverage, but it may result in thick, non-isotropic voxel and slice misregistration due to the motion. In contrast, 3D TOF acquires volumetric data across the entire slab, offering high spatial resolution, isotropic vectors for multiplanar reformation, and a high signal and a high SNR. However, it is limited to small converging areas, slightly longer imaging times, and is more sensitively to motion artifacts, which can affect the entire accuracy sense. So we simulated an equation with 4 parameters. The T1 and T2 parameters are used for different blood type. In this video, we use the arterial blood. For the TR value, the signal intensity will dramatically increase when TR increase. And the max point of the curve will shift to the right, which means the best flip angle for intensity will increase. And for the TE value, we can say when the TE value increase, the signal intensity will drop little bit. But the max point of the curve will not shift to the left or the right. So that means TR will be the Cohen parameter decide the best flip angle. And we also do the 2 figures for arterial blood and venous blood. For arterial blood has a smaller T1 and bigger T2 compared to the venous blood. With the fixed TR and TE, the arterial blood will have better signal intensity than venous blood. That explains even TOF reduced TR for arterial blood due to the faster blood flow rate, the image can still contain good intensity. Since TR will be the Cohen parameter, describe the best flip angle, we try to see the max difference between the blood and green matter and the blood and the white matter, which refer to data 1 and data 2. So data 1, data 2 located a point that with different TR, the max signal difference will at which location on the curve. We can see the max difference for both green matter and white matter is not at the highest point of the curve, but they near the highest points. So even we pick the angle that max the contrast between blood and matters, it will still contain a very good signal intensity. And another result we found is that when we increase the TR, the gap between the data 1 and data 2 will increase. That means the flip angle difference will increase when dealing with different matters near blood. To maximum contrast, it is not advisable to simply increase TR to achieve high signal intensity.
So in conclusion, in our presentation, we summarize the principles of TOF imaging, particularly for MRA or MR angiography. And we also highlighted its non-invasive nature, its clinical applications. We explored the role of one key parameter, which was the flip angle, and how it can optimize signal contrast between flowing blood and stationary tissues. And finally, we used our research to develop a MATLAB simulation, which demonstrated the effects of several other parameters, especially for arterial and venous blood signals. And finally, we can say in conclusion that TOF imaging is a critical tool which can be used in detecting and monitoring several vascular abnormalities. Thank you.
Thank you. Any questions from here for the few people remaining? Okay. I have a question. So can you explain, did your simulations take into account the velocity of blood?
No, we didn't. We take it down like 4 different parameters, but we didn't consider the flow of the blood.
Okay, so the difference between gray matter and white matter was simply based on the difference in T2 star?
Yeah, T1 and T2.
T1 and T2, okay. Okay, great, thanks. All right, well, if there's no other questions, we're gonna end this session. So congrats to everyone and have a good break. Good luck with your finals if you still have them. For those of you submitting your video presentations, don't forget they're due today. So let's stop.